Distinguished participants, ladies and gentlemen, very good morning to all of you and welcome back to the third IUFRO Acacia Webinar Conference from Kuching, Sarawak, Malaysia. Ladies and gentlemen, we have now come to the second day of the conference, and this morning we will have the privilege to have the Director of Forest Sarawak, Yang Berbahagia Datuk Hamdan bin Haji Muhammad, in delivering the second keynote address for the conference. Then we will continue with the third session, which is risk management, followed by the fourth session on high value added timber product. Without any further delay, it is now my pleasure to invite again Professor Dr. Wickanaswari Ratnam as the moderator for the keynote address sessions.
Thank you, MC.
It is now my pleasure to invite again Professor Dr.
Very good morning to all. Welcome back.
For the keynote address.
Hello, hi Vicky.
Hi. There's some noises in the background, sorry. So I shall start with the introduction of our second keynote speaker, Datuk Hamdan. Datuk Hamdan bin Haji Muhammad, the Director of Forest Sarawak, holds a Master's degree in Environment Science in Land Use. Our second keynote speaker, Datuk Hamdan. Datuk Hamdan bin Haji Muhammad, the Director of Forest Sarawak, holds the Master's degree in Environment Science in Land Use. Our second keynote speaker. Sorry, I think I was unmute. Sorry. As Director of Forest, sorry for not, I don't know from where I was muted. So I just continue. In year 2019, Datuk Hamdan was promoted as Director of Forest, Sarawak. Sorry for not, I don't know from where I was muted. So I just continue. In 2019, Datuk Hamdan is actively involved in forestry sector in Malaysia, particularly in Sarawak. He has been nominated as a board of honorary advisors of Sarawak Timber Association, Mutual Sendirian Berhad, board member of Hardwood Timber Sendirian Berhad, Sarawak, Planted Forest Sarawak Forestry Corporation, Sarawak Forestry Corporation Sendirian Berhad, Lubuk Kiara Sendirian Berhad, or Lembaga Amanah Masjid Negeri Sarawak, member of Sarawak Diversity Biodiversity Council, permanent member of Forest Research Institute Malaysia FRIM from 2018 to 2022, as well as steering committee member of Sarawak Civil Service. Datuk Hamdan's significant contributions to civil service and forestry have led him being awarded the Meritorious Service Medal Award, which is a silver award, in year 2008. Officer of the Order of the Star of Hornbill, Sarawak in 2016, and Order of Meritorious Service to Sarawak, which brought him the title of Datu in 2019. With its progressive and forward-thinking, the department is transforming towards the new FDS by fostering new people, teams into achieving its vision to be globally recognized in sustainable forest management. Without much further ado, I present to you all Datu Hamdan.
Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Thank you to the session moderator for your kind introductions. Ladies and gentlemen, today we have come to the second days of the conference. Indeed, it is such a great pleasure and a very honored to be part of this conference and to present a keynote address entitled Forest Plantations Towards Sustainability of Wood Productions and Wood-Based Industry in Sarawak. These are the presentations outlines that I will highlight on my presentations today. Before I proceed, allow me to briefly show the locations of Sarawak State in Malaysia, as shown in these world maps. Sarawak is one of the three regions in Malaysia which are located on the northwest Borneo Islands and is bordered by Brunei Darussalam and Kalimantan of Indonesia. Sarawak covers an area of approximately 12.4 million hectares, and it is the largest state among the 13 states within Malaysia. Sarawak is known as one of the 25 biological hotspots, which means a place that's found to have the highest diversity of trees anywhere on Earth. Out of 12.4 million hectares of total land mass, 62% is still under forest cover. The management of forest land in Sarawak comprised of 3.9 million hectares of permanent forest estate, 0.8 million hectares of totally protected areas, and 2.8 million hectares of the statland forest. Permanent forest estate has been in existence as early as in 1920s. While the main functions is for timber productions, permanent forest estate is also a very important area for water catchment, source of livelihoods for local community, biodiversity conservation, and other ecosystem services that provide for the socio-economic prosperities of people of Sarawak. As of now, we have 3.9 million hectares with numbers of 117 permanent forest estates. This is our department's efforts and with the support from relevant agencies to achieve the 6 million hectare target area for gazetted permanent forest estate. Yes, it is a long and challenging process, but the journey will definitely continue. Nowadays, the timber industry especially has been known as one of the key contributors in the state development and socio-economic growth, with the log productions from the natural forest peak in 1991 with 19.4 million cubic meters. This has shown a great progress since the state government began to embark on plantation forest development in 1997 with the aims to supplement the productions from natural forests. In terms of governance, forestry sectors has been well regulated through Forest Ordinance 1958. Thereafter, subsequent amendments were made in 1996 and 2015 for the licensing process, and issuance will abide with the rules and regulations. As for the planted forests, this Planted Forest Rule 1997 and Nursery Rule 2017 were the governing legislation. Meanwhile, on research and development, effort has started since 1920s with trial planting for local growing species. Thereafter, in year 1970s, focus was emphasized on both local and fast-growing species. Reforestation of degraded area, such as certifications area, has started with local and fast-growing of exotic species. Sarawak has targeted to achieve 1 million hectares of planted forests by 2025. Planted forests play an increasing role in ensuring that sustainable forest management policy are achieved in Sarawak. They provide an alternative source of raw material to meet the increasing demands from wood industries and thereby relieve the pressure from the nature forests. In these slides are the forest plantations policy objective adapted and modified from both 1997 and 2020 versions. The revised policy directions on industrial forest plantations endorsed by our Chief Minister of Sarawak, who is also the Minister of Urban Development and Natural Resources on the 22nd of January 2020. Amongst the highlighted are, number one, to ensure sustainability of sufficient raw material for timber industry in the state. Number two, to relieve the pressure of logging over the natural forest. Number three, planted forest has shorter rotations, which is 5 to 10 years compared to natural forest, which is 20 to 30 years cutting cycle. Number four, planted forest is expected to have higher wood yields of 150 cubic meter per hectare per rotations compared to natural forest of merely 45 cubic meter hectare for cutting cycle. And lastly, the state government has set a target of 1 million hectares of planted forest by 2025, with the aim to produce 15 million cubic meters of the log per year. Forestry sectors has been well regulated in terms of governance. The first one was brought about through Forest Ordinance 1958, and thereafter subsequently amendments in 1995 and 2015. For the licensing process and issuance will abide with the rule and regulations. As the planted forest, this Forest Rule 1997 and Nursery Rule 2017 were the governing legislations. The total area of licensed planted forest in Sarawak, with a gross area of about 2.1 million hectares. Map showing the distributions of the industrial tree plantation in Sarawak, where the biggest concentrations falls within the central regions of Sarawak. Under revised policy directions for planted forest 2020, Sarawak government has targeted to certify all eligible area by year 2025. Sarawak regions accounted for the biggest areas certified under planted forest. There are 6 forest plantation management units certified under MTCS PAFC's Malaysian Timber Certification Scheme, with the total areas about 95,434 hectares. This table shows the progress and achievements of our planted forest, which is 521,908 hectares as of now. This graph illustrates the planting progress in hectares as June 2021, with about 521,000 hectares area planted with various species, which in green lines compared to our targeted in red dotted lines. Production from planted forests has steadily increased over the period of 10 years, from 4,726 cubic meters in 2011 to about 1.7 million cubic meters in 2020. The harvesting of log productions from planted forests was decreased in 2020 and 2021 due to the global COVID-19 pandemic. The total revenue collected from our planted forest timber from 2011 till 2021 is about $25.8 million US. The top 3 major species planted were acacia, made up the bulk planted species at 55%, followed by Falcataria molucana, 21%, eucalyptus, 12%, and Neolamarkia cadamba, 7%, and other species, 6%. Next few slides, we show some photos of the main species planted in Sarawak. This is samples of acacia species of plantations. This one is a Eucalyptus pelita photo, mainly planted in Bintulu regions of Sarawak, Falkataria, Malukana, also locally known as Patai, Kelampayan, Onio Lamarkia Kadamba. Monitoring and auditing activities to ensure planting progress being carried out according to the tree planting plan submitted and the compliance to the relevant rules and regulations. Amongst the activities involved are field verifications or ground truthing versus half-year report submitted by the license holders, compliant audit to the legal provisions, engagement sessions with the plantation's license holders through meeting, dialogue, seminar, and other kinds of platforms, and industrial forest plantations data management. Auditing on progress plantings are being carried out using advanced technologies such as satellite imageries to interpret data image that shows planted and non-planted areas. Helicopter monitoring for air surveillance, especially at our remote area, and the ground truthing for the site verifications and counter-checking of reported progress. This slide just to show some harvesting activities at our planted forest in Sarawak. Currently, there are two harvesting methods being practiced by the licensees: cable yarding and shovel system. These are some of the end use of the tree from planted forests such as solok, veneer, wood chip, medium density fiberboards, pulp and paper, and pellets. These pictures also show some product from acacia timber, displaying both indoor and outdoor furniture. Another picture showing the furniture product, some example of house structure, bench, dining tables and chair, panels and floorings, and many others. Under the concept of new generation plantations, research and development should be intensified to produce the optimum growth of tree through better genetic improvement programs in order to get the best growth, resilient species, and feasible returns to the investment. There are more emerging genetic improvements programs coming in view with more research efforts and collaborations to facilitate aggressive plantation development of the state. Some of the issues and challenges faced by the stakeholders in Sarawak in ensuring a robust growth of the planted forest industry, both upstream and downstreams, are namely, number one, geographical difficult terrains, remote areas, contributing to the costly establishment and management of the plantations area. Number two, R&D that is still initial stage with incoordinate research conducted by different entities, either government or the private sectors. Number three, lack of R&D expertise in a critical upstream area such as tree improvements or breedings, silviculture, soils, site species matching, integrated pest and disease, and management. R&D at the downstream level, namely high value added product, in particular engineered wood structure, biocomposites, and etc., and lacking of effective mechanism for small stakeholder participations in the expanding the planted forest area. Before I conclude, there are the way forward as it has been agreed under the revised policy directions on industrial forest plantation 2020. Close monitoring of planting progress, applications of advanced technology and best practices for planted forest management, intensifying R&D for high productivities, mandatory certifications on the eligible area for planted forest management certifications by 2025. Develop a new deal for For example, agroforestry program for dispute plantable area with the native people based on the agreed partnership model. Distinguished guests, ladies and gentlemen, forest plantation is the solutions toward the sustainability of wood productions to supplement the demands of our wood-based industry. Looking at our past trends in the forestry sectors, there is an urgency to address the widening of supply and demand gaps in the upstream and downstream of our timber industry. Thus, the successful implementations of the planted forests would lessen the harvesting pressures on our natural forests and promote the sustainability of our forest resources. Although our natural forests are highly productive in terms of biomass productions, commercial timber productivity is very low, which is at about 2 to 2.5 cubic meter hectare per year, because most of the tree has no commercial timber value that must be reserved for protection functions of the environment. Forest plantations, on the other hand, can be extremely productive if we assume a conservative growth rate of 30 cubic meter hectare per year. The annual production of 30 million cubic meter from our planted forest can be produced from plantation area of about 1 billion hectare. The current timber productions from our natural forest now is only 3 million cubic meter. In other words, the future supply of timber from our planted forest is 10 times what we have produced currently. Forest plantation is to release pressure on harvesting in our natural forests in terms of area intensity. These forest plantations yielding timber, logging in area is needed for protection function will substantially decrease and may be phased out. The successful implementations of the planted forest would lessen harvesting pressure on the natural forest and subsequently would enhance sustainability of the natural forest and reduce negative environmental effect. Distinguished guests, ladies and gentlemen, with that, I end my presentation here. Thank you for listening.
Thank you very much, Dr. Hamdan, for the most excellent overview of forest plantation and sustainable production of wood in Sarawak. We are not taking any questions for this keynote. So I will now hand over to the Secretariat, please.
Thank you again to Professor Dr. Wiknoswari, and special thank you to Yang Berbahagia Dato' Hamdan bin Haji Muhammad. Despite his busy schedule, he managed to be here with us, delivering his thoughts and insight, especially the way forward in transforming forest plantations in Sarawak in line with Sarawak government, as well with global industry needs and demand. Distinguished participants, ladies and gentlemen, moving right along, I am more pleased to welcome our moderator for our next sessions on risk management, Professor T.S. Dr. Kalima Abdan. She is currently the director at the Institute of Tropical and Forestry and Forest Product, INTROP, University Putra Malaysia, Higher Education Center of Excellent Tropical Wood and Fiber, Ministry of Higher Education's Malaysia. She obtained her bachelor degree in agricultural engineering from University Putra Malaysia in 1996, and she obtained her PhD in biocomposite technology also from University Putra Malaysia in 2006. Her interest is biocomposite manufacturing technology, textile composite, and disaster management research. Ladies and gentlemen, please welcome Professor T.S. Dr. Kalima Abdan.
Thank you very much for the kind introductions by the MC. I'm Kalina Abdan here, your moderator for the next section for the third IUFRO Acacia Conference 2021 in the scope of embracing transformations of sustainable management of industrial forest plantation, with the scope specifically on managing the risks on acacia plantation. I hope that everyone can hear me very well. Is it good? Can I proceed? Give time to the Secretariat to welcome three invited speakers for today's session. With the first session will be delivered by Dr. Anto Rimbawanto, Principal Researcher Scientist, Center for Forest Biotechnology and Tree Improvement, CFBTI Indonesia, with the title Challenge of Pulpwood Plantation in Response to Pests and Disease Threats. The second invited speaker will be Mike Wingfield, Pest and Disease Specialist, BFC Research and Development, Sdn Bhd, South Africa. And the third one is Stuart Fraser, Research Group Leader, SciCon, United Kingdom. Okay, now we do have the slide is ready for Dr. Anto Rimbawanto. Allow me to read his CV. Okay. Dr. Anto Rimbawanto is a principal research scientist in Center for Forest Biotechnology and Tree Improvement, Indonesia, and has been working there since 1994. He then obtained his PhD from Australian National University, Australia. His research interest covers a range of topics related to forest tree breeding, molecular breeding, conservation of genetic resources, population dynamics, DNA log tracking, species barcoding, biosecurity, pest and disease in plantation, and the range of species including Acacia mangium and the rest of the species that covers on his expertise. Since 2001, has been the project leader for several IUFRO Acacia projects on managing disease of pulpwood plantation in Indonesia. Without further delay, is it Dr. Anto Rimbawanto ready for his speech?
Yes, I'm ready.
Yeah, please welcome. The floor is yours.
Thank you, Professor Kalina. Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Good morning, everybody. It's glad to be here, and I'd like to thank the organizing committee for inviting me to this very important third Acacia conference and give me the opportunity to share our experience. As mentioned in the short CV, I've been working on Acacia project to solve acacia diseases, and in this opportunity, I'd like to share our experience as well as presenting some of the project findings. As I learned over the last 15 years, finding a cure for forestry diseases is a race against time. New diseases or disease emerges before we completely comprehend the conditions and how to control it. Yes, so this is just a story of an Acacia project, and it was presented or prepared together with my colleagues from University of Tasmania, Professor Caroline Mohammed and Dr. Morik Plant. Next slide, please. Thank you. Pulpwood plantations is a vital industry for Indonesia, and it's supporting an industry that covers productions of up to millions of tons of pulp and paper. Roughly, there are about 1.5 million hectares of monoculture plantations been established to support the pulp and paper industry. And Indonesia is positioned in the 10 world biggest pulp and paper producers in the world. Every year, Indonesia produces about 16 million tons of paper and 11 million tons of pulp. and the industry generate an export value of up to 7.2 billion US dollars, an increase of 15% on annual basis. It's also a very important industry, providing workforce of up to 500,000 people, and indirectly about 1.2 million people involved in this industry. Next slide, please. Plantations is basically dominated by two genera, acacia and eucalyptus. And acacia became the choice of species because when it was introduced to Sumatra in the 1970s, it was quickly proven to be a suitable species for alang-alang grassland. And, therefore, when the industrial plantations was launched in the 1990s, Acacia mangium was the apparent choice for the industry. And soon genetic improvement was carried out, and up until now, after a series of rotations, a cycle of selections and breeding, we managed to increase the productivity of operational plantations from 20 to 47 cubic meter per hectare per year. So it's quite significant achievement in terms of breeding program. We also learned from this experience that breeding can only start in earnest when large plantations of the species is being established. And this is the good examples of Acacia mangium and Eucalyptus pallida later on of the breeding program. For about 2 rotations, the plantations managers have had a good honeymoon with the plantations being pest and disease free, but soon after, disease becomes a daily routine of their management. And at some stage, disease even caused some significant loss for the plantations company. Acacia mangium is dominating the mineral soils in Sumatra and Kalimantan, and on peatland, which is also a large proportion of the area is being planted with Acacia crassicarpa, as this is the species of choice as for the conditions of wet and low pH conditions with the soil. Although I must underline that the productivity is much less than Acacia mangium. Now, because of disease and pests, the Acacia mangium has gone from the landscape of plantations in Indonesia. More than 600 hectares of Acacia mangium has been replaced by Eucalyptus pallida, although the number may be higher. For those of you who are familiar with the industrial plantations in Indonesia in the 1990s, the popular pictures of Acacia mangium plantations was the one planted in South Sumatra. That photo is no longer a popular pictures anymore because it's been replaced by Eucalyptus pallida. Next slide, please. Good. Now, incidentally, a book on tropical acacia was written by Dr. Ken Oates and colleagues in 2000, which identifies a significant disease in acacia. Acacia started to support a plantation disease project in Indonesia in 2001, being implemented collaboratively between University of Tasmania and the Center for Forest Biotechnology and Tree Improvement, and focus on addressing the hard rot on acacia. But as we conducted the field survey to assess the damage of hard rot, we found that root rot is much more damaging and expanding quite quickly. So subsequently, our research focus shifted to root rot caused by Ganoderma. In the book, Ken and colleagues listed a number of diseases there, which was already exist, but it's not so damaging. Things like root rot, ceratocystis, and and leaf rust. And in the pest side, Helopeltis was also identified as one of the problems. Now, due to the mortality of growth rates of Acacia mangum, which was hardly obtained from a series of breeding program, has been reduced to less than 15 cubic meters per hectare per year. While in non-infected areas, the growth rate is still maintaining quite high. So, in other words, the hard work of breeding is lost because of the pest and disease. That's why, as underlined by our keynote speaker yesterday, Dr. Sadana Nambia, that we have to focus also on how to breed species which is tolerant or resistance to pest and disease. Next slide, please. It has become apparent that Acacia mangum is highly susceptible to this Ganoderma root rot disease. With increasing rotations, the intensity of the damage is also increased. In our survey, we recorded between 3 to 28% of root incidents. And in subsequent recording, it has increased to more than 35%. So it's as the inoculum built up in the plantations, the intensity of damage is also increased. There are several Ganoderma species that are been identified, but Ganoderma philippii and Stereotanum has been the main pathogen in Central Sumatra, in South Sumatra and Kalimantan, as well as in Central Java, causing widespread damage. Our simulations back in 2016 estimated that if research on root rot can mitigate losses by only 1%, this is equivalent to at least 4.2 million per annum at the current price. So it's quite significant that genetic or control of root rot was very important. But as I mentioned, as we'll mention later on, while we are still trying to understand the root rot problem or find the solution for root rot problem, new diseases, which is much more damaging, has occurred. Next slide, please. These are the species that have been identified during the project using an ITS sequencing. At least 6 species have been found in Acacia and eucalyptus, but the 2 major ones was Ganoderma philippii and Ganoderma stereotanum. Next slide, please. In the field, it's easy to identify when we do the survey assessing the damage of root rot. We will look at the concentrically expanding patches of dead trees or disease gaps. We can look at trees that had chlorotic and thin crowns, such as indications of infections. And when we expose the roots, we look for some reddish brown rhizomorphic skins on the root, as on the pictures on photo C. And also we can look at the myceliums under the bark of infected fruit body, infected roots. Sometimes when we were lucky, we can find the fruit bodies appear at the basal stem under some infections, and this can be used for identifications of species, but more often than not, the fruit body is not there. So that's why much quicker identification of species using the NAITS sequencing is used in the project. Next slide, please. Now, as I mentioned earlier, that while we are trying to understand the Ganoderma, a new disease become widespread, which is caused by ceratocystis. It's caused wilting on the trees, and it died quite quickly. This is a widespread disease, which has a wide range of hosts, not only in trees but also in fruit and other tree species. and it's been reported in a number of countries, including Brazil, South Africa, as well as in Indonesia. Study found that there are different species of Ceratocystis, but the one most present in Indonesia is Ceratocystis manginicans and Ceratocystis acacia. Next slide, please. And this is the symptoms of the disease. Those of you working with acacia are quite familiar with this. Colleagues in Vietnam have also had the same problem. It's called wilting and death of the trees. It has a bark discoloration lesions on the trees and gum exudations and stem cancer. When you go into the plantations of acacia, which is infected by Ceratocystis, you've got an acid smells coming out from there. And it's an indication that there's been an infection on the trees. Because there's also an insect hole from bark beetle, an insect factor. Interestingly, there's a widespread damage of monkey in Acacia mangium plantation in South Sumatra at the MHP plantation site, and that's become a trigger for the widespread damage by Ceratocystis. So a combination between pest and disease has become a major cause of the destructions of Acacia mangiums in South Sumatra. It has a specific order. There are pictures of Dr. Budi Cahyono from Smart Forestry. He's one of the experts in plant diseases in plantations in Indonesia. Next slide, please. Now, while we are working on Ganoderma, the only solution for controlling the Ganoderma is through biocontrol agents, because genetically there is not much room for finding a resistance or tolerance individual of Acacia mangium. Therefore, we go into the approach of developing a biocontrol agent using fungus that was found in the field. For quite a while, we developed this, identifying the suitable fungus as well as the method of deploying the biological control in the field. Next slide, please. This is a series of work that was carried out. We found that two biological controls that are effective to controlling this Ganoderma, which is Plebeiopsis species, and Serena. These are the two major fungus that is found to be antagonistic to Ganoderma. So we did a series of study to assess the biological control activity of the fungus and how the BCA competes for space and nutrients in petri dish, as well as we did a test out in the field, which is going to be presented in the next slide, please. Next slide, please. Okay. So this is how we apply the biocontrol agents. We prepare solutions with inoculums of biocontrol and then applied to different states of the conditions there in the stems, so there's newly freshly cut stems. And then following that applications, then we will dig out the roots and find if there are BCA presence in the root system. So it's quite a series of work they're being done to assess the effectiveness of the BCA. In summary, we were able to present that BCA can, although this is still early, BCA can suppress the growth of Ganoderma but we're still continuing the assessment to see how soon the BCA can destroy the stems, because this is also an important source of inoculum. As soon as the stem is gone, then the inoculum will also disappear. Next slide, please. Yes. So this is the one that we developed with Ceratocystis. We developing a disease resistance of tolerant clones. Through the series of selections of tolerance, we've been able to select up to 15 or 20 clones of resistant or tolerant Acacia mangum clones, which is now being planted and secured in a clone bank for further assessment. And later on, one of the keynote speakers, Dr. Ari, will present the performance of this resistance Acacia mangum clones for the Ceratocystis disease. Next slide, please. So again, this is the only solution. If you're going to continue planting Acacia mangums in the future, you have to find a tolerant Ceratocystis Acacia mangum or hybrid tolerance to Ceratocystis, which is also being carried out in Sabah softwood, for instance. Similar approach being done here, and the hybrid seems to be more tolerant than the pure Acacia mangum. And at the time being, as we haven't finished the work, the screening was focused on Acacia mangum. So this has also become a part of the parameters of breeding in plantation company in Indonesia, that screening has to be part of the breeding program, has to be screened for tolerance to pests and diseases. Otherwise, all the hard work of breeding will be lost before it was harvestable. Next slide, please. So I think, yes, next slide. I think the management strategies is quite obvious here, that an integrated approach has to be carried out, including biological control, breeding for resistance, and also plant health, hygienic plant silviculture practices also has to be part of the integrated approach. By this kind of approach, I think we can control the disease of pests in a manageable level. Next slide, please. Well, this is a new project that we are going to carry out from 2021 to 2024, 25. A continuing project from what we achieved in the past, as well as developing a new work, particularly in the modeling of pests and disease in the light of climate change. This is also happening in our plantations, and we will also use early detections of biotic damage using unmanned aerial visualizations, as well as continuing the breeding work focused on hybrid, which is being carried out at the moment. And also strengthening forest biosecurity networks in Southeast Asia. And this project will be carried out between Indonesia, Vietnam, and Australia, the University of Tasmania. So with that, I'd like to conclude my presentation. Next slide, please, and express my acknowledgements to various organizations which have involved in this project, and particularly to Australian government for the funding support, the University of Tasmania for technical, as well as collaborations between plantations companies in Indonesia Sinar Forsi, the Jalan Tol, Musi Hutan Persada, and the University of Gajah Mada in Yogyakarta. Thank you very much for your attention, and Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you, Dr. Antorin Bawanto. Thank you for his nice presentation for the most valuable information on pest and disease management strategies. Those information on biological control with several detection procedure applied and its status of breeding for the resistance. Okay, without further delay, there is no question for the first invited speaker. We're going to compile it towards the end of the session, and then we go for the next presenter, Mr. I try to read his short bio. I think, yeah, Professor Mike is ready. Okay, Mike Greenfield, founder director of the Forestry and Agricultural Biotechnology Institute, FABI, served as an advisor to the University of Pretoria Executive. His research focuses on the health of forestry globally, having published widely on this topic, together with more than 100 past PhD students from many countries of the world. He served a 5-year term as president of the International Union. Sorry, okay. He served as 5-year term as president of the International Union of the Forest Research Organization, IUFRO, and currently a past president of 2924. He advised forestry companies and academics organizations globally, particularly in terms of promoting research excellence and maintaining healthy forests. He has been widely recognized for his research, including receiving the Nkrumah Science Award from the African Union and being awarded honorary doctorate from the University of British Columbia, Canada, and North Carolina State University, USA. Okay, we most welcome you, Professor, and to deliver your speech.
Kalina, thank you very much, and good morning to everybody. I wonder if you could take off, I'm going to share my screen, so if you could just take off the slide, I'll share my screen.
Okay, sure.
Thank you. This thing.
Yeah, we can see your slide. Proceed.
Let's see if I can see it. Sorry, that you should be able to see my slides now. And sorry for that slightly slow start. So thank you very much for your kind introduction. It's wonderful to be with you. I'm sitting in a very early morning, I think something like 3:30 a.m. in Stockholm, where I've been able to enjoy the wonderful Marcus Wallenberg Foundation ceremony. My talk, I think, follows very, very nicely against the background of our previous talk by Dr. Anto, and I'm going to be focusing, as you've already said, on Ceratocystis wilt of acacia and our efforts to understand the disease and to reduce its impact. So, to really get down to the beginning of the story, this disease, and unfortunately not a lot has been published about it, but the disease appeared first in 2003 in Indonesia. And this picture is from those days in Indonesia long ago, where I was in a situation where I could see a provenance trial that had been pruned, so it's Acacia mangium that had been pruned, and trees were wilting. This is quite shocking, and of course we didn't know what the cause of the disease was, and as already been said, there was knowledge of Ganoderma root rot in the area, reasonably serious, but nothing like what we were seeing emerging. We started work trying to identify the pathogen, the cause of the disease, and recognized that it was a Ceratocystis species. And that led a number of years later to the really fundamental work done by Martin Turrigan, who started a master's degree, who completed a master's degree studying this disease. So Martin really should be credited as giving us the first solid information as to the cause of this disease that started emerging, well, in 2003, or perhaps before that, we just hadn't seen it. Let me share with you a few pictures of the symptoms and how this disease developed. And Dr. Anto has already given you some background. So this picture is in fact from Sarawak, where this meeting is being hosted. And these trees are older trees. They would be in the region of 5 or 6 years old that are dying. So what started to happen relatively rapidly is that young trees were dying usually one would see this in the first 6 months to a year, but older trees were also started to die. And there are two different disease dynamics that drive the death of older trees and younger trees. Although today we don't see the problem with older trees because there just aren't any left in most areas that I've worked in. The disease developed very rapidly, first in Indonesia, to a point where very large areas of trees were dying, and they became unprofitable to continue to propagate. So fell to waste was unfortunately the consequence. And really something quite dramatic, and maybe amongst the many diseases that I've worked with in forest plantations around the world, one of the worst. The symptoms were typical, of course, of Ceratocystis disease. And what you see here, and it's important to look towards the outside of this section through a tree. The tree is not dead yet, it's still green, but if one saw the whole tree, it would be a wilted tree. You see this streaking discoloration, and that is because the pathogen, so the Ceratocystis, moves in the vascular tissue of the trees. And that's really important to the whole biology and understanding this disease. So it moves through the vascular tissue, causing the streaked discoloration. And one wants to always look towards the outside of these sections to see the streaking. This is perhaps one of, to me at least, one of the most interesting aspects of the disease. When one peels away the bark of a tree that's just showing early symptoms, one sees these very distinct lens-shaped lesions. And if one reads back into the literature on Ceratocystis diseases, that is actually quite a common symptom. What is happening, and I'd like you to imagine this, is the pathogen moving inside the vascular tissue of relatively large trees. The pathogen is, of course, a fungus and aerobic, so it's jumping out to the bark to, of course, get oxygen, so coming out. But what happens here is the bark around these small lens-shaped lesions then is dry and pops open, and the fungus, and this is something special about Ceratocystis, produces a very strong fruity odor. So it is able to produce its spores and grow on the outside of these lesions, and that attracts insects to these lens-shaped lesions. So it's quite special to the biology of many Ceratocystis species. I call this my Ceratocystis flower. So peeling away the bark of the trees, one sees these dramatic lesions. If you're a pathologist, you'd say it's a very beautiful picture, but of course our trees are becoming diseased, which is not ideal. So the first step in dealing with this problem was to identify the pathogen. Now, as I've already said, Martin Tarragon did some fundamental work here. Those were the days where we had access to fewer genes to sequence, less genetic information to identify Ceratocystis species. Today, our tools are much more powerful. But clearly, a Ceratocystis species, we would, for identification, wish to isolate it. So here again is one of these lens-shaped lesions. You see in the smaller picture these very distinct fruiting bodies, ascomata or perithecia to some that are here, and the asexual state is this white fluffy stuff here. So to isolate the pathogen, relatively easy, picking up spores from these structures and putting them into agar, and then to sequence genes. This is a tree produced, in fact, for the paper for this meeting by Irene Barnes, with some of the Malaysian isolates, Indonesian isolates, and then isolates from mango in Oman. And I won't go into the mango story, but this is a 4-gene tree, but a very distinct species of Ceratocystis. And Irene Barnes will talk a little bit about that. I think there's another talk as well, talking about the identification of the pathogen and why it's different to, for instance, the pathogen that infects sweet potato. A key question for us, of course, was to identify how this pathogen is spreading. And I'll say right up front, and Dr. Anto referred a little to this, but I think a really important issue is we know very little. Scientifically, there's a lot of anecdotal data, but there are very few strong quantitative studies that have been peer-reviewed. So not a lot quantitatively known about how this pathogen spreads. And in fact, some of our own observations are only that they're observations, they're strong observations, but there's a great need for deep research to better understand this disease. So we first looked at knitted beetles. Now, for those of you who don't know insects so well, knitted beetles are commonly called picnic beetles, and that's because they tend to visit people having picnics where there's fruit and fruity aromas around. They're attracted to these fruity aromas, and the fruity aromas from ceratocystis, of course, are going to attract them. But our interest in knitted beetles also, of course, relates to an understanding of ceratocystis. So there's a great body of literature pertaining to knitted beetles and their association with ceratocystis. So, of course, that would be a logical place to look. So this is a picture of a tree that is beginning to die. They commonly have this foamy exudate. That foamy exudate is not ceratocystis. It's mainly yeasts and bacteria that are building up inside these dying trees. There's a lot of pressure, and they kind of pop out, and they pop out where the ceratocystis crack the bark open or where there are insect holes. But that yeasty foam is incredibly attractive to knitted beetles, and you'll see little black spots here. These are knitted beetles. The next picture will show that more clearly. Here are knitted beetles in this foam, and their larvae are here. So this is how the knitted beetles breed. They lay their eggs, and the larvae develop in this foam. And of course, they will pick up ceratocystis species. So a great deal of work was done in trying to understand the role of knitted beetles. So some of our team, authors of this presentation, putting out pineapple traps and other traps to catch knitted beetles. And I'll say right up front that we were very unsuccessful, at least I was hoping to get thousands of beetles and lots of ceratocystis. We have been able to collect knitted beetles. This is a selective approach, not really a medium, to isolating ceratocystis. We use carrots. It's a well-known baiting technique, and these little bits here are knitted beetles are full of ceratocystis. So what I can say is we've been able to connect knitted beetles to the ceratocystis problem, but there really are no solid data. So I'm talking about large numbers and quantitative data. So the exact role that these beetles are playing remains unknown. They're involved, but we don't know how. Or how deeply. That takes us to the scalitine. So these are coleopteran wood borers. They are very common. As trees start to die, they become, and Dr. Anto also referred to this, they become very heavily infested by these little scalitine bark beetles. There are two key species from the Malaysian work that we've done, Eualacia perbrevis and Xyloborus crassiasculus. So looking at those a little more, isn't this a fabulous picture? Here you see these beetle holes. So there's the entry holes of the beetles. They're going into the ceratocystis infected tissue. The little lesions around them. So we have spent a lot of time looking at them. They will not go into dead wood. They go into trees that are starting to show symptoms of death. They're very clever. You can see their holes here. But it's very common to find them associated with dead branches because one has dead tissue here. There's stress, there's ceratocystis, and then they go in around that area. So one sees a dead branch here. This is still green tissue, and there's a hole that one is going into. And this illustrates the same very nicely. Ceratocystis in the wood and lots of beetles around the branch area. So that's where they are most attracted in the beginning. So we spent a great deal of time catching these beetles. There's a beetle trap, you can see an emergence trap. Again, team members trying to understand what's going on there. This is many people that work on these beetles call these noodles. So this is the frass, the dust that the beetles produce. And this dust is coming out of a tree that's infected with ceratocystis. So what we can do is we can collect that frass and put it onto carrots or a selective medium and isolate ceratocystis quite easily. But that's not surprising because that tree is full of ceratocystis. So think about a beetle going in there, pushing out the bark, and out comes frass or dust full of ceratocystis. The issue is how important are those beetles in terms of vectorship? So this is an interesting picture, I think. So what we have here is fresh cambium on a tree, and what's happened here is an elephant has pulled off the bark of the tree. So nice and fresh. And a few days later, we see all these lens-shaped lesions. Look at them carefully, and there is the ceratocystis. And right on the surface is frass. So I have no question that frass from these beetles is part of the dispersal of this pathogen, probably a huge part of the dispersal of the pathogen to any wounds. And it's important to remember here that ceratocystis is, they are wound infecting pathogens. They're very aggressive pathogens in many cases, but they need wounds to infect. So any wound, and we've already heard the issue of monkey damage, elephant damage, squirrel damage, all incredibly important. And I agree with Dr. Anto that in the beginning of this problem, so going back to 2003, and as the problem became increasingly worse, animal damage, monkey damage was really important to the development of what became a massive epidemic, has become a massive epidemic. So the next issue is to look at soil-borne propagules. The reason we thought about soil is that some ceratocystis species, think about the sweet potato pathogen, are soil-borne. They're well adapted to live in the soil. And a question was raised as to whether we didn't actually have a root problem. So could this be a root disease? So we've looked, this work incidentally has started in 2013 with the Borneo Forestry Cooperative. So it's a 9-year project that I'm presenting. But there was a lot of discussion about the role of soil-borne pathogens and whether it couldn't be in the soil. So we, of course, have done huge surveys to look at look for symptoms in the soil and have never found any evidence that this has got anything to do with root infections in terms of the disease. But we looked at the potential of soil-borne propagules. Here's Redzwan Raf, also an author on this presentation. And the way we did this is went into an area with very heavy ceratocystis infection. We know it's ceratocystis infection because all of these trees have been inoculated with ceratocystis, and all the spaces are where trees have died and they're still dying here. So to take soil from an area with a very heavy potential inoculum of the ceratocystis and isolate from that. And thus far, we've not been able to isolate ceratocystis from any of that soil, which to me is even strange. I would have expected to find some ceratocystis, even though it may not be involved in the disease. But so far, we've not managed to find ceratocystis in the soil. So I don't think a soil phase has anything to do with this disease. Then to establish a reliable inoculation protocol has been incredibly important. And we've heard a little bit about inoculation of trees. So a big question for us was, what is an appropriate age to inoculate trees? So we know that this pathogen is a vascular well pathogen, and it requires, if we want to understand it clearly, it requires a well-developed xylem system, so a vascular system. And to think about that a little bit, what we've done is we've inoculated, we've taken clones that we know have high levels of resistance, and I'll come to that in a minute. A small number of clones inoculated, so these are clones that we would expect to be resistant, inoculated them, and within a few weeks, they're all dead. Same here, so these are 2 clones, about 2, Redzwan Raf has done this work together with us. Within a couple of weeks of inoculation, these hopefully resistant clones are dead. Here is one that is a control, just to show a comparison. So we're convinced that inoculating small trees is pointless, and I'll go to that a little later. So the inoculum and the inoculation procedure is really important. While it's easy to isolate the fungus, one wants to be working with isolates that are highly virulent. So we've gone through a process of inoculating many trees with a number of isolates and selecting highly aggressive isolates, and then maintaining their aggressiveness through every year, putting them through trees in successive inoculations and re-isolating. So our first set of field inoculations, and this was published, Jeremy Bronner, who's speaking in this webinar, was involved here. This work was published. We inoculated 6-month-old trees. And here you can see an inoculation site. What happens very rapidly within 6 weeks, the trees are dying, very aggressive development. And what we realized that 6-month-old trees in the field are inordinately susceptible and really don't give us solid results. This is just the same thing. You can see these incredible lesions, very, very long lesions in the tree. So what we decided at that point, and as I say, this is a 9-year project, so it's gone on for a very long period of time, is to inoculate only 1-year-old trees. So here is one of the early trials. Some of the authors online here will remember block 104C. Many, many dead trees. So what we found is to inoculate a year-old tree with a single point inoculation, about a 0.5 centimeter size piece of agar with fungus, one single point, and just to leave the trees and observe them. And what happens is most of them die. Some of them grow quite well for a while and then fall over. You can see a single healthy tree here. And there's been a process of then capturing by cloning these very few, very small numbers of trees for further work, for verification. So here's an aerial photograph, a drone photograph showing a block where we've inoculated trees. And you can see how few trees, these are inoculated at a year old and just left to die, evaluated a year later or so, and one sees a very small percentage, and that'll be about 5% of the trees, 4 or 5% that remain. And those can then be cloned and tested, and Mahathir will talk, Lapamo will talk later in this symposium on the verification process. So growing those trees to large trees again and verifying that they actually are resistant. This is just a picture of the same sort of thing. Another of these blocks, you see some nice eucalyptus alongside here that have been inoculated for 1 year and then left and evaluated over time. This is a picture from a seed orchard. In fact, these trees, so this was a breeding seed orchard of trees that we hoped had resistance but were never tested. They were inoculated as large trees and they mostly also died, very small numbers, didn't die from the inoculation and have been captured in clones. So that then gets us to the way forward. And I'm at 18 minutes now, so I need to finish up. So Dr. Anto talked a little bit about the future and Acacia auriculiformis. And that really, I believe, also is where the future lies. So we have done some work, this picture taken in Sabah forest industry plantations, and thanks to them for helping us with this work. So these were, this trial was Acacia auriculiformis mangium hybrid. You see trees here inoculated also at about a year old. These have died, but those have not. So there are clearly clones of the hybrid that are highly tolerant to infection by Ceratocystis. The real question is, these trees in the front, of course, is what one doesn't want. So how do we avoid that? And I believe the answer lies in Acacia mangium. So what we have now is trees that it's a small number, unfortunately, because there's very little resistance in Acacia mangium to Ceratocystis. But what I believe we have is we have trees that are highly tolerant. They're not perfect, but they are highly tolerant. They're from clonal material now, through large-scale field inoculations, then verification after that again in the field, that we can now use in breeding and selection for disease tolerance. Another piece of the future, and you've already heard this, is lies. So this is a picture from Indonesia going back many years. This is all mangium that has collapsed and died due to Ceratocystis, and one sees the beautiful eucalyptus in the foreground. Here's a helicopter. I'm sitting in the helicopter photographing out, and one sees the future, the amount of the future that lies in eucalyptus plantations. So Eucalyptus pellita has been very successful in this area. And is being planted increasingly, and that is something to really feel positive about as we develop acacia for planting, because I believe that we have opportunities for acacia, particularly the hybrid in the future, and we should pursue that aggressively. What we need is really good research. The caveat here, of course, is that Ceratocystis is emerging as a disease of eucalyptus. So you can see a pellita plantation here. This is an SSB or Sabah softwoods. A dying tree here, typical Ceratocystis symptoms. So we're going to have to be very careful, but I'm very positive about eucalyptus. It's very different to acacia in the sense that we have many opportunities to hybridize, to select, and to clone. It's a much different process. So I think a large part of the future is going to be Ceratocystis and other disease tolerant eucalyptus in the area. So that gets me then to the conclusions, and I'm a minute over, for which I apologize. Over a 9-year period, important aspects of the biology of Ceratocystis manginecans have been determined. Infections are typical of a canker wilt disease without evidence of any root association. The pathogen is spread in the frass of wood borers and is apparently spread by nitidulid beetles. Inoculations of small plants do not reflect the field situation, and screening must be done on established, at least 1-year-old field established trees. We've now inoculated 6,000 trees, these established trees in the field representing 140 families. And this has led to the identification of a small number of highly disease tolerant clonal trees. So we're really excited about that. And disease-tolerant clones represent a major step forward for the acacia forestry and can now be used in future breeding programs. And this is especially, I believe, with Acacia auriculiformis, which has shown high levels of tolerance to infection. So in conclusion, we've had a great deal of fun, and I want to thank many colleagues and friends, not only the authors of this presentation, but just many colleagues in Indonesia who've contributed very substantially to the work. 9 years of work, a lot of activity in the field, and you can see just a few pictures of us in meetings or busy enjoying, but also studying a very complex disease. And then, of course, there's a team in South Africa that I've led in the past, and numerous of these young people have been involved in the molecular genetic work that has gone behind the work that I've presented today. So I thank you so much for your attention. It's wonderful to be with you, and I'll look forward to the question session a little later. And again, I'm 2 minutes over, so apologies for being a little slow.
Thank you very much, Mike, for a very nice presentation and insightful information on the ceratocitis, on the disease and the effort to reduce its impacts. All the procedures, inoculation and column, should be applied at the seed, has been applied at the seed occurred, and we can see that there is some improvement, and the acacia hybrid that we can hope that it can able to fight the disease. And the last slide on the eucalyptus promising plantation tree. Okay, we go for the next presenter later. Before that, is it Mr. Stuart ready here? We do have one question, but we put it towards the end of the session. Okay, Mrs. Wat, you are ready. Allow me to read your biography, please. Okay, Sir Fraser is a forest pathologist and a research group leader for ecology and environment at SciCon. His research focuses on the ecology, epidemiology, and management of forest disease. He leads and contributes to interdisciplinary projects that aim to improve the resilience of productive and natural forests. Prior to joining SciCon, Stuart undertook a postdoctoral fellowship on an economically important of rust disease on Acacia mensi at the Forestry and Agricultural Biotechnology Institute, FABI, University of Pretoria, South Africa. Without further delay, the session is yours. Please, Dr. Stuart.
Great. All right, I'll just share my screen. Hopefully that's showing. And hopefully that's showing. So could I have a signal that that's working?
It's working. We can see it.
Oh, awesome. Perfect. Fantastic. Cool. Yeah, so I'm going to be, thanks for that introduction. Yeah, I'm a forest pathologist at Scion, but I'm going to be talking about work I did during a postdoc at FABI in South Africa with Mike, Yolanda Rue and Alistair McTaggart on the life cycle and field epidemiology of the rust pathogen Euromycladium acaciae, which is a major problem on Acacia mearnsii in southern Africa. So just a bit of an overview of the talk. I'll give a bit of an intro to Acacia mearnsii. I'll talk about the emergence of the disease as a problem, and then I'll talk about what I actually did in the postdoc, which was work looking at the optimum conditions for infection by the pathogen, development of a resistance screening protocol, work looking at the life cycle and field epidemiology of the pathogen, and then identification of a mycoparasite commonly associated with the rust, and just wrap up with some kind of conclusions on that work. Acacia mearnsii, or black wattle, is a species native to Southeast Australia, but has been grown in South Africa. Can you see the top of my slides, or? I'll just keep going.
See them.
Yeah, okay. So yeah, it's grown in South Africa, over 100,000 hectares in South Africa, mostly kind of smaller scale, grown by farmers. It's grown for pulp from the timber and tannin production from the bark. It contributes largely to the South African economy and employs a lot of people. So it's an important species. A rust species, Euromycladium, was first found on the species on the acacia in the 1980s, and this paper was written about that by Mike and the two Mikes. This was an asexual form of the rust, so these are spores which are called Uridinia spores, and these are the asexual form of the rust. And it was present in the 80s. It wasn't causing much damage. It caused a bit of defoliation of the pinnules, but didn't cause too much damage. And this was the only life stage of the rust which was found. Then later, in I think 2013, kind of significant damage was found, and a significant disease was found. So as well as the uridinial rust, so the asexual rust, which had these spores, there was new spore types were found. So there was this teleospores, which are sexual spores, were found associated with the damage, and these produced these are the spores here, these kind of dark brown masses, and under wet conditions they oozed out of telea and matted pinnules and other parts of the plants. So this was causing kind of significant damage. And before I arrived, Alistair McTaggart and the rest of the team did some genetic work and found that based on a phylogenetic species hypothesis that this was one species. So the Euromycladium, the Uridinia, the asexual form, and the sexual form, which was now causing the damage, was one and the same thing. So just some photos here of kind of the symptomology. So again, this is the slime that's produced by the spore masses under wet conditions. These kind of can coat the pinnules and cause defoliation. They can affect all kind of above parts of the plant, so the leaves, the pinnules, the seed pods here, the flowers, the stems, and it can also cause deformity here. So, and in this, I don't know if you can see me, but here we have a kind of exclusion trial on a young plantation here. This has got a fungicide applied, so no rust here, and you can see the rust having an impact up here. So this was quite a new behavior. So the kind of one of the main questions when I arrived was this a change in behavior of one species, or was this a kind of new introduction of a very closely related species? The other things that we wanted to look at were that very little is known about these species. They're very understudied group of rusts. So very little was known. So we also wanted to identify the optimal conditions for the rust, for infection. We wanted to develop an artificial inoculation protocol for resistance screening, so we could speed up resistance breeding. We wanted to elucidate the life cycle, like I said, and kind of look at whether it is one species or two species or one or two lineages, perhaps. Whether it was a change in behavior. We also wanted to look at kind of field epidemiology, so the seasonality of spore release, infection, symptom development, those kinds of things. And whilst we were doing that, we detected we observed a mycoparasite commonly on the rust, so we wanted to identify that and look at the seasonality of that as well. So just to kick off, I'll talk about the work we did looking at the optimum conditions for infection. So I did quite a bit of work looking at germination of spores. So I kind of concentrated on the teliospores as they were the main spores associated with the significant damage. And I looked at optimal conditions for their germination, and they germinate, produce this basidia, and then they produce another spore type, these are called basidiospores. And these are the actual spore type that could then infect the acacia. So we did some germination trials, and they kind of gave us a head start, and then we quickly moved on to looking at infection of live plants. And what we found was, in terms of temperature, 15 degrees was optimal for infection, and then a dew period, so a wetness period of 24 to 48 hours was optimal. We had very little infection at higher temperatures at 25 degrees. And the other important thing to note was that only the very youngest growth or tissues were susceptible to infection. So these older, fully developed leaves were not susceptible to infection. So we took that information and we developed a artificial inoculation protocol, which we published. And to do that, we did a proof of concept trial with 12 different families of Acacia mearnsii. We took 12 that they'd been looked at in the field and putatively identified as more resistant or less resistant, more susceptible. So you can see these 12 different these 12 different families, variation within each family, but also variation between families. So we had more resistant families and more susceptible families. What was good to see was that here we've got the artificial inoculation scores going up on this axis, and here the field rust scores. The ones that we identified as more resistant in the artificial inoculation were also more resistant in the field. So that was very promising. And I think we'll hear more from Julie and Moreno-chan, who I did this work with tomorrow, which might cover some of this work. So the next thing I want to talk about is some of the field work we did, looking at the life cycle and the field epidemiology of the disease. So here's the proposed life cycle of the rust from Alistair McTaggart's work, which was published in 2015. And here, originally, like I said, in the 80s, and from the 80s on until about 2013, the Uridinia and Uridiniospores was the only stage known. So this is the asexual stage, which just can cycle. It can infect, produce Uridinia, and reinfect. From 2013, spermagonia were also noted. Telia were also noted, and these telia, as I showed, they germinate, or they spread, then they germinate, they produce these basidiospores, these other spore types, which then infect, produce the spermagonia. And the hypothesis was that then they produced Uridinia, and then the Uridinia produced Telia, which is the kind of typical ontogenic rust life cycle. So that was what was hypothesized by McTaggart. But in my work, what we saw in the artificial inoculations I've just mentioned was we never saw Uridinia. So we inoculated with the basidiospores, we had spermagonia develop, and then that transitioned straight to Telia. So we didn't see this at all. So that kind of built towards this idea that perhaps there was two different rusts present, and that this new highly damaging teleal rust is actually a new introduction. So to look at this, and then also to look at the field epidemiology, so that the timing of symptom development and spore release, we did monitoring at 3 plantations on the eastern border of South Africa with Eswatini. We did this between January 2016 and April 2017. We did monthly disease monitoring and weekly spore traps with these quite cheap and basic spore traps, which are just slides coated with Vaseline. And we also recorded temperature, relative humidity, hourly, and rainfall weekly. Here you can see some of the symptoms that we were monitoring, and here's some of the spores on the Vaseline, which were counted. So first up, in terms of the life cycle, we took collections of infected material every month, and we looked at the different life stages of the rust. And looked at how they co-occurred. And what's interesting to note here is how it kind of varied across time. Here we've got spermagonia and telia, and you see that they were co-occurring regularly and frequently. In the hypothesized life cycle proposed by Alistair McTaggart, spermagonia would have transitioned to uridynia, but we never saw spermagonia and uridynia in close association without telia. So this lends itself to the idea that these are two kind of separate rusts. Then the thing that we also did was we had some difficulty doing, or I had some difficulty doing molecular work with this rust. It was quite challenging. But looking at one region of the genome, what we found was uridynial rust, so the kind of original 1980s asexual rust, was different to the telial rust in one position. So we had just the uridynial rust in the Western Cape and up in Limpopo, but we had both life stages in these other areas. And but what we can see is that there was a kind of fixed difference in this region. Again, kind of supporting the hypothesis that we have, if not two species, two separate rusts, but very closely related rusts. And that's what's shown on this slide. We've got the new what's probably a new introduction causing most of the damage, which is a telial rust called Uromycladium acaciae. And this is what I saw in my inoculations, and this is typically what we saw in the field. And the other Uromycladium species or lineage is just uridynia, but they're very, very closely related, and they share a most recent common ancestor. So following a phylogenetic species concept, we can't separate them. And what we really need is a study in the kind of the populations of origin in the native range in Australia to look at this further. So what we also did from that monitoring, the stuff that, like I said, we did the spore trapping to look at the timing of spore release to kind of give us an idea of when, if we were going to use chemical intervention, for instance, when it might be best to do that. And here we've got the year. So starting in July, so this is midwinter, running through to June. And in the area where acacia is grown, we have dry winters and wetter, warmer summers. So you can see that in terms of temperatures. Up in the summer, in the around January, and this is also when rainfall and humidity peaks. And what we saw was we got an early peak in teliospore production and release in October kind of time, and then we had a major peak kind of towards the summer season. And then so we looked at the relationship of spore release, of teliospore release with weather variables, and kind of unsurprisingly, we found a significant relationship with rainfall. So we got a peak at the 2 sites where we kind of got most spores. We had a peak in detections at about 5 millimeters of rainfall. So that's at this site, and at this site, a similar pattern. At this site, we didn't see the pattern, but we had far fewer spores. And then similarly with relative humidity, we saw an increase in detection of spores at increased relative humidities. So this indicates that the spores disperse in water splash, and this is hardly surprising looking at this kind of slimy, the slime that's kind of produced by these spores under wet conditions. So you can see they have this kind of fascicle that comes off the spores, and this bursts under wet conditions and kind of creates this sliminess. And then when it dries, you kind of get this crust of spores, which kind of protects the spores underneath. So hardly surprising to see that it likely does spread in water splash. And then just the last bit of my talk, we found Sphaerolopsis macroconidialis really commonly on the Euromycladium acaciae. So this is a hyperparasite, so a parasite that attacks the rust, and this was a new host and country record for this species. And what we found, because we looked at the seasonal occurrence of this microparasite, and what we saw was this peaked in abundance in the kind of winter and springtime. So outside of remember from that previous graph, this is when the epidemic of the rust occurs. So we kind of get it mostly affects outside of the main epidemic period. But it might have an impact on the overwinter survival of the rust, and may lower inoculum at the start of the year. So it'd be interesting to do some more work on that microparasite. So just some conclusions and then some acknowledgements to wrap up. So we found that 15 to 20 degrees with 24 hours to 48 hours of wetness was optimal for infection. And that we took this information to develop an artificial inoculation protocol for resistance screening, and that's available for breeders to use for the breeding program. Resistant families were, or more resistant families, should I say, were identified, and they had we found a good relationship between the artificial inoculations and field trials. There's likely 2 taxa or lineages of Euromycladium of the rust present on Acacia mearnsii in South Africa. But if we wanted to separate those out, we'd have to do much more work in the native range of the rust. The damaging teleal rust, which was first kind of noted in 2013, is likely a new introduction to South Africa. Teleospores were mainly released in our study in the second half of the wet season, and they're likely spread by water splash or under wet conditions. And we identified the microparasite Sphaerolopsis macroconidialis on the rust, and this might reduce the overwintering of the rust. So lots of acknowledgements. People at Fabi, so Louise Shuey, worked with lots of people at the ICFR, so Ilaria, Tabili, Julian, and Andrew, and then worked really closely with industry and with some international collaborators. So, and thank you very much for listening.
Thank you very much. Start on your insightful presentations on the life cycle of the rust pathogens on Euromycladium acacia. So we can find that a positive and polynomial relationship between all the factors that have been measured. And then we believe that this insightful information could help on the find the better risk and mitigations for the disease and solutions. Okay, we are ready for the question and answer session now. I'm receiving few questions. The first two questions, I believe that already have been DM to Mike, or should able to read the question, Mike? The first question from Lucy Tian. Yeah, please, Mike.
Do you want to just read the questions? There's so many of them here. So the one question I saw was from Simon Lawson. He asked a question about the vectorship and the beetles and whether there is any obligate association with the beetles and Ceratocystis. So there are no Ceratocystis species that have obligate relationships with wood-boring beetles. There are related fungi that do, but there are no examples of boring beetles that have an obligate association with Ceratocystis. So what I was saying is, it seems to be that the major role of the boring beetles is in producing frass. So they're going into trees that are infected, producing a huge amount of frass that is infected with Ceratocystis, and that's blowing. I mean, you can just, I think we're probably breathing in inoculum, and that's blowing around the plantation area. And if there are any wounds, they could be monkey wounds or squirrel wounds or pruning wounds, they become infected. But I'll make another point, and that is that we have very, very little solid experimental data for this disease, even though we've known the disease for, well, since 2003. And I was listening to Stuart's talk, and it's nice to see you, Stuart, far away from where I am. You know, just listening to your talk and seeing the depth of experimentation that has been done on the rust disease that you studied, we really don't have strong, solid data. I saw a question, I think it might have been directly to me from Dr. Nambiar, and thank you very much for that. And he asked a question about the situation in Vietnam and why it seems that the Ceratocystis disease is not has not been as dramatic in Vietnam as, for instance, in Malaysia and Indonesia. And I can think of three things. Firstly, we don't know a lot. So, you know, solid quantitative data is not really available. But the disease seems to have started primarily in Indonesia and appeared later in Malaysia, if that's in fact correct, and then in Vietnam. So Vietnam may be behind this curve. The other thing is Vietnamese forestry and foresters have pursued the hybrid option for a long time and done some very fabulous work on hybridizing Mangium and Auriculiformis, which is clearly more tolerant. So I think that's a piece. The other thing that we know is we've done quite a lot of population genetic, and we is a big we. Of course, Irene Barnes, who will talk later, has driven quite a lot of this work. We've done a lot of work on the population genetics of this pathogen, and it's genetically very variable, particularly in Vietnam. So there might also be differences in what's happening there. And it does remind me, if you don't mind me making the point, that I saw that I left out my acknowledgement slide, which acknowledged a lot of wonderful people who have been involved in this work for a very long time. So I apologize for that, and that can maybe be shared at some point later. But I think that's enough from me. Let's look at other questions.
There is a question from Lucky Tian, wanting Dr. Mike mention about pathogen spread through insect and soil. Do you come across of airborne spread of Ceratocystis in plantation?
Yeah, Helena, thank you for reading that and tracking these questions. So I think I've already said that in the sense that there appears to be a lot of, and we again don't have quantitative data, and that's the problem. We're trying to get it, but you know, what one needs is directed, like master's, PhD studies on each of these particular questions. But we know, and you would have seen that from my presentation, that the frass of beetles is very heavily contaminated, and there's a massive frass in the air, is very heavily contaminated with Ceratocystis and probably some other fungi as well. Kira Lin will talk later, not on this particular disease, but you know, she's been doing some work in that direction. So there for sure is a lot of airborne inoculum. And I think that is probably the major driving force. But we also think that other arthropods might be involved. So there are many more questions than answers. Now, again, I'll make the point that I think I believe that we need very much more detailed research on this disease, but also on other diseases, insect problems, in terms of forestry and the humid tropics. I think that's already been said. And I was in an interesting discussion in the last couple of days with colleagues who were making the point that the way that we have fought, the world has fought the COVID pandemic, has relied deeply on solid, very solid, basic, fundamental research that has been done around the world for many, many years. And unfortunately, we really lack that sort of depth of knowledge in terms of the problems that we're dealing with.
Hello.
There's Budi.
Thank you, Professor Wingfield, for your excellent presentation. I learned a lot from your presentation. Recently, we found in our eucalyptus pelita plantation that some Ceratocystis also infected one of our clone that previously very resistant to this eucalyptus, to this Ceratocystis. And we found that this happened after we have a lot of herbicide drift. So what your comment on this?
Well, firstly, Dr. Budi, it's lovely to see you again. I just wish we were all meeting in person. So let me make two comments. Ceratocystis is appearing in eucalyptus more and more. We don't know too much about it, but the one thing we do know is that there is a lot of genetic variability in the Ceratocystis. So looking in population genetic work that we're doing, and we have genomes of many isolates today of this pathogen, there's a lot of genetic variability. We're coming to understand that, but we must also understand that isolates will differ in their aggressiveness to eucalyptus, to acacia as well, for that matter. So the pathogen is able to change, probably change more rapidly than we would like to believe. Think again about COVID and the change of the SARS-CoV-2 virus. Fungi change quite quickly, and so the answer to your question could be two things. One is change of the pathogen. So you have and we have many examples worldwide in plantation forestry of resistant clones becoming susceptible, and usually that has to do with a change in the pathogen. The connection to herbicide herbicide damage is a very difficult one to answer, because it would suggest then that maybe herbicide is stressing trees. Now, my experience with Ceratocystis is that the trees do not like, that the pathogen does not like stressed trees. It likes young, green, vigorously growing trees. So if we stress trees with herbicide and then did, for instance, an inoculation, I think we would find less damage. So I would be inclined not to connect herbicide damage to this problem, but rather, if I were taking a guess, I would say change in the pathogen so that resistant clones are becoming susceptible with time. And I think that's crucially important as we go ahead into the future, to remember that the pathogen is changing, and that trees that are tolerant and selected for tolerance today are not necessarily going to be tolerant in years to come. So I would put my money on a change in the pathogen and not a connection to herbicide. But again,
thank you for your comment, Prof. Thank you.
It's a pleasure.
Okay, Mike, there is another questions in the Q&A, but from Sri Sunarti. Have you found that questions?
I can't. I've got a long list of comments and hellos and goodbyes, and inform a little bit more about the tolerance of acacia to Ceratocystis. What kind of hybrid is it? Okay, so I'm not sure I fully understand the question, but thank you for opening up this issue of disease tolerance and the hybrid. So what we know is that where hybrid Acacia mangium auriculiformis has been inoculated, or even through natural infections, there seems to be a much high level of tolerance. My feeling is that most of the hybrid has not, so the hybrid parents have not been tested for disease tolerance. So what I'm saying is we have huge opportunity for hybrid, but what we need to do is we need to start with mangium that we know has high levels of tolerance and auriculiformis that we know has high levels of tolerance. I can tell you that I with colleagues from the Borneo Forestry Cooperative over the years, and thanks again to them that were left out in my acknowledgement, not my acknowledgement slide, or the one that I didn't show, but we've been out to very old trials of pure auriculiformis and found that some of those trees are starting to die. They probably grew very well until the pathogen appeared. So that's evidence that the pathogen came later, appeared later than the planting of those trees. But those trees would not have been tested for tolerance to infection. So in some ways, I think we almost need to go back to square one. Now for mangium, we've done that. We have numerous clones that have high levels of tolerance, but we need to breed with trees that are of known tolerance. And again, I make the point that the only way to do this is not to be tempted by quick fixes and trying to inoculate little plants, but get to establish trees, inoculate the trees, select tolerant trees for auriculiformis as well, and then do the breeding. So I think that's the only way forward, but I also believe that it's a huge opportunity for forestry in the humid tropics, because acacia is a fabulous tree. I see Dr. Nambiar I can now see him, which is really nice. It's good to see you. I'm a very, very strong supporter of the future of acacia forestry. We just have some challenges, and those are good for us.
That's great answer, and then good discussion among from Mike and the team. And what else we can have here is to Dr. Anto. Would you mind to answer the question? Have you found the questions? I can assist you by give you the direct message. You are muted. Would you mind to choose on?
Yes, I got it, Professor Kalina. I think, yes, it's questions about acacia hybrid that from Cincinnati. This is my colleague. He's part of the member of the Asia project here. But the questions is quite involved in the hybridizations of acacia, mangium, and auriculiformis. Questions is, should we develop the F2 acacia hybrid, even though the variations is wider than F1 and the productivity information is still limited? Yes, I think we have to, it's important that we focus on, as mentioned by Professor Mike Wingfield, that focus will be first on the tolerance and resistance of this acacia hybrids to the disease, because if we find that F2 hybrids is much more tolerance than the F1, then that's the way we should go. Productivity is something from this experience of dealing with disease. Productivity should be a second priority after we manage to control the disease, because mentioned earlier with Pabudi in Eucalyptus plantae, and pointed out quite nicely by Professor Mike Wingfield that the pathogens evolve quite rapidly, and the tree species is left far behind. So as soon as you've got a individual that is resistant, tolerance to ceratocystis, when you deploy it in the plantations, the ceratocystis pathogen has mutated or somewhat. It's typical similar to the Delta variants of COVID-19. We're still trying to cope with it, and who knows, maybe another variant will emerge pretty soon. So I think my preference will be focus on selecting the tolerance and then improving the productivity at some stage later on. That's probably what I could say to answer that, Kalina. Thank you.
Sorry. Thank you, Dr. Anto. There is one question has been DM'd to you. Would you mind to read, have a look?
Is that who's that question to?
Is that for to Stuart?
Will one resistant clone of Mangium be susceptible to infection by different Ceratocystis species? I think Dr.
sorry,
Anto,
that one for Mike. Yeah. I have
could have been
another question. Yep,
to
please,
either
Mike.
of us, but I think Dr. Anto has actually answered that question already. There are other Ceratocystis species. There are many moving around the world, and others might appear. But I think we need to be most focused on manganecans and it changing. So as Dr. Anto said, it has huge capacity to change. It has a sexual state, and there's already a huge amount of variability, genetic variability in the pathogens. So I think we must expect that our tolerant material, and that would be hybrid material, is going to collapse. And we're going. This is with pests and diseases. This is an arms race. So and that's with the tree pests and pathogens, human pathogens. It's the same. It's all the same thing. The day you stop working, then all problems appear. So we've got to continue to do research and be ahead of the diseases. And there, I'll make the point that we're doing a lot of work through the Borneo Forestry Cooperative now, looking at eucalyptus, Pelita, and the Pelita hybrids. Also work being done in Indonesia and other projects. And I know that there are others online here that are involved in that work. But there are numerous new diseases appearing, not just Ceratocystis, but other diseases appearing in the eucalyptus forests. We'll hear talks later. We see pathogens like the rust pathogen emerging, Teratosferia destructans getting really bad in some areas. So it really is for pests and diseases, an arms race, and we just have to continue to study and be ahead of the problems. And that's lots of research and lots of genetics. And I think that's what we lack, perhaps, is strong, detailed research on these individual problems, as opposed to relatively peripheral observational work, which is important, but we need data. We really lack data. Again, I see Stuart is nodding his head, but I think of the amount of data on that one disease, Stuart, that you worked on. And that's the kind of thing that we need for many of these problems.
Okay, thank you, Mike. Stuart, do you receive your email? Sorry, receive the question?
Yeah, so
Okay.
yeah, thanks for your question. So it's, can I elaborate more on how the knowledge of the biology of the rust pathogen contributes to development of strategies for disease control? So I guess, so the first part is hopefully of the talk was probably hopefully quite straightforward in that understanding how optimum conditions for infection, that kind of thing, that we took that through to the development of an artificial inoculation protocol that could be used for resistance breeding and could significantly increase the speed of resistance breeding. So I think it would be important to keep doing field trials as well, but I think the use of the protocol could quickly increase that. In terms of the other things, I guess it's in terms of the life cycle, it's really to know, are certain stages important or not, I suppose, going forward, and should we focus on some more than others? The other thing is it something that's changed, or is it a new introduction? And if it's a new introduction, well, how did it get in, and how can we block that pathway in future? So that's really important. Then the other thing in terms of spore release mechanisms and timing, it's kind of, I guess, chemical use isn't used too widely, but it was being used in South Africa in the acacia plantations when I was there. And you really, if you want to have an optimized chemical control operation, you really need to know when spores are released, when infections are happening, and you really want to know as well in terms of management, in terms of spread. If it is spread mostly by water splash, that would kind of have implications for perhaps spacing plantations and things like that. So it comes back to this talk about COVID. You know, the more you know, the more you can do, really, I suppose.
Okay, thank you, Stuart, and thank you, Mike, and thank you, Anto Rimawanto. I think we have to end our session. We have to give a line for our session to post the session, a virtual tour, and then we meet again at 11:10. Thank you very much for all the invited speaker, a very insightful presentation, very informative, and then I believe that all the participants had their very great information and their feedback from the question and answer. There is one more question for Wagner Morris to Mike, but I wish that Mike can answer it by type the answer better. Thank you, everyone, and have a good day ahead. And we meet again at 11:10. See you again.
You see that, Mason?
Mm.
It says on Sarawak, the Sarawak disease is not so. Distinguished participants, ladies and gentlemen, we will have a tea break sessions here. So we will start again in 11 and 10. Continue with the risk management sessions again with Dr. Kalima. So see you all again as mentioned by Dr. Kalima. Thank you. moderator for this risk management sessions. Thank you. Dr. Kalima?
Thank you, thank you, MC. Okay, we start again our session this morning. I would like to ensure that Mr. Nampan ready? Okay, you are there. Okay, and I see. Nampan is a PhD candidate at Forestry and Agricultural Biotechnology Institute, PABI, UC of Pretoria. He is trained a forest pathologist with a strong background in molecular biology. His research focuses on different aspects of plant pathogenic fungi, including taxonomy, population biology, genetics, and genomics. He specialized in identification of disease, characterization of causal agents of tree disease, and elucidating of the epidemiological of the pathogen and the disease they cause. He is particularly interested in shoot and foliar disease that are important to the forestry industry, especially in the southern hemisphere. Okay, Mr. Nampai. Okay, looks like you're ready.
Yeah.
Okay.
I don't know if I hope everybody can hear me clearly. Yeah.
Yeah, we can hear you well.
Okay, let me try to share my screen.
Okay.
I don't know if this go through.
Yeah.
Okay.
Yeah, it's perfect. Okay, please. The floor is yours.
Okay, thank you. Good morning, everybody. My name is Namfa, and I'm a member of the Forestry and Agriculture Biotechnology Institute of FABI from South Africa. So it's my pleasure this year to present at the IUFRO Acacia conference. And today my talk is about a new and serious disease called eucalyptus scab and shoot malformations, and as an emerging threat to the plantation forestry in Southeast Asia. So as we all know, Southeast Asia has fast-growing and extensive plantation forestry programs, and they are generally short rotation, monocultural colonial plantations. And the plantations of eucalyptus species has been established rapidly in Southeast Asia, importantly also to replace the acacia plantations that have been severely damaged by canker and wilt caused by Ceratocytis marginicans, as presented previously by Brookmire Wingfield. However, as this eucalyptus plantation expands, the threat from pests and diseases also grows. As being said, in 2014, a new and serious leaf and shoot disease of unknown etiology appeared in eucalyptus plantation of North Sumatra, Indonesia. This unusual disease was unlike any other foliar or shoot disease known on this tree elsewhere in the world. The disease first appeared in a small plantation area in the Lake Toba regions of North Sumatra, Indonesia. Since its first appearance, the disease has become widespread in the affected area, covering approximately 40,000 hectares of eucalyptus. The disease is strongly linked to environmental factors, especially temperature and the rain. Disease symptoms appear shortly after the onset of rain, and trees begin to recover at the onset of dry periods. We have conducted an experiment to see the effect of rain splash on spreading the disease by building an overhanging shelter to cover the trees inside from the rain. The result were indeed shocking, where the tree inside were completely healthy, as you can see here, and while the one outside of the root got severely infected. So what is the typical symptom of the disease eucalyptus scab and shoot malformation, or ESSM? The disease is characterized by black necrotic spots that appear initially on young leaves and petioles, which then become scab-like as the lesion age. Spot can be scattered like on the first photo, or concentrated like on the second photo, where rainwater accumulates on the leaf surface. This eventually leads to deformation of the leaves, causing curvature usually on one of the leaf lobes. In severely infected tree, lesions can lead to girdle, crinkle, and distort shoots and leaves. In some of the most susceptible clones, secondary symptoms include a feathering effect, resulting in loss of apical dominance and abnormal elongated branches. Severely infected clones usually die after a number of successive infection cycles. So using normal light microscope, it was impossible to observe any visible fruiting structure or any fungal structure. But when we use scanning electron microscopy or SEM to observe the few collected symptoms, we found that there was evidence of fungal spore associated with the scape-like structures. Effort has been made since 2014 trying to isolate from the disease materials using culture-dependent methods, where we have obtained hundreds of fungal isolates. The fungi were grouped and identified based on morphology, as well as DNA sequencing for correct identifications. As you can see from the graph, we have obtained members of more than 30 fungal families associated with the disease symptoms, some of which contain well-known group of plant pathogens. However, the fungal community from the disease symptom was too diverse to the point that we could not pinpoint what could be the primary pathogen of the disease. We then went a further step to use culture-independent methods, in this case amplicon sequencing, with the hope to have a better idea regarding the community associated with the disease without the need of obtaining pure fungal cultures. We extract the DNA from the disease and the healthy material, and then compare the microbial community in those samples. And we found an increase in the activity of different groups of Basidiomycetes yeast, as well as Ascomycetes yeast in the disease material collected from the field. Based on that results from the next generation sequencing, we then try to culture and characterize the yeast community associated with the disease symptoms. We found out that the yeast community were again very diverse, and they belong to more than 10 different families of both Basidiomycetes yeast and Ascomycetes yeast. We have done some pathogenicity trial with the representative yeast far, but none of the inoculation with the yeast show similar symptoms like from the field. The yeast diversity were perhaps the consequences of the infection, but not really the causal agent of the unknown disease. All the unsuccessful effort make us realize that we need to focus again on the isolation strategy. The isolation needs to be done more precise, focused directly on the infection points. And we again pay more effort in isolating more from disease symptom and expect and look for the candidate to be something more consistent between sampling periods, different sampling periods. So in 2020, after a long time trying, we have managed to obtain another set of isolate before the global shutdown due to COVID-19. This isolate immediately caught our attention. They usually emerge from the disease tissue fragment after 5 to 7 days and grew extremely slowly on media. And all the isolate have distant dark red to brown raised colonies. The colonies also produce a distant dark to dark red pigment that diffuse into the grow medium after a while. And when we look at the fungal community isolated in the past, we found that we have collected some isolate with similar morphology before. In fact, it was only 4 isolate out of 1,000 of isolate collected in the past. And they belong to the family Alcinoaceae. Initial blood result of the ITS show that those isolates reside in the genus Alcinoae, which is known for causing what is called scape and spot anthracnose on plants, usually horticulture and agriculture crops. We took all the isolate and sequenced 4 different gene regions. Phylogenetic analysis of RTS, LSU, RPB2, and elongation factor individually, as well as in combination of these 4 gene regions, consistently revealed that those isolates collected from North Sumatra represent a distant clade, and these were clearly separated from other previously described species in Elsinoi. So in fact, we are dealing with something completely new and never been found before. So to confirm the pathogenicity of the putative causal fungus, we have used the isolate and done the inoculation on eucalyptus leaf material under laboratory conditions. Amazingly, all the isolate tested for pathogenicity on detached eucalyptus grandis branches produce necrotic spots absolutely similar to those found under field conditions, only after a short period of time, like 5 to 6 days after inoculations. And after 14 days, the spot dry up and turn dark brown at the center and become scab-like. The species of Elsinoi were consistently re-isolated from the necrotic spots on the inoculated leaves. And this infection were consistently absent from the negative control as well. We again use SAM to further confirm the infection from the inoculation trial. The scanning electron micrographs of the infections on inoculated leaves after 14 days show that the necrotic spots were identical to those under field conditions. And fungal structure, like hyphae continuogenous cell or conidia, were obvious at the center of the spots, and they were these were similar to those seen from the same images from the naturally infected tissue as well. So after approximately 6 years of focused research, the unusual disease had been now formally characterized and named as eucalyptus scab and shoot malformations. And the causal agent were described as a new species called Elsinoi necatri. The name was chosen to refer to the destructive impact that the disease has on the eucalyptus host plant. So the discovery of Elsinoi necatri causing serious and important disease in the plantation forestry environment is the first time a species of Elsinoi has been found on non-native eucalyptus hosts. So what could be the possible origin of the pathogen? An intriguing question that has significant relevance in terms of eucalyptus plantation development in this region and also globally. The first possibility is that the pathogen could have emerged from native metaceae that are common in Sumatra close to the plantation area where the disease occurs. It could be similar to the situation with many other eucalyptus pathogens, such as species of Chrysopathy, or the myrtle rust pathogen, Australbuccinii cdi in South America. Alternatively, the pathogen might have been accidentally introduced into North Sumatra, where it had encountered susceptible host tree and an environment conducive to infection. Such an interaction could have come from the area of origins of eucalyptus, including mainly Australia and nearby Indonesian islands. So to be able to answer such question of the origin of Elsinoi necatri, we use a genomic approach to investigate further. So 2 genomes were sequenced for 2 Elsinoi necatri isolates. We then used these genomes to characterize the mating type locus of Elsinoi necatri and develop the primer set that can distinguish between different mating type isolates, as well as a set of polymorphic microsatellite markers. So we will be able to investigate the reproductive biology of Elsinoi necatri, the genetic structure of the population in North Sumatra, and also to answer the question of does sexual recombination occur in these regions, and hopefully can understand the possible origins of Elsinoi necatri. So now back to the plantations. So few observations have shown that there is evidence of differences in the susceptibility of different eucalyptus clone to infection of Alcinoid negatrix. This suggested that there is variation in the host susceptibility and provides us the opportunities to control the disease through active breeding and selection for disease tolerance. A rapid screening technique is now being established to effectively identify disease-tolerant planting stock in order to reduce the significant negative impact of this new and serious disease to the forestry industry in this region. The system is now being optimized. We can be able to screen for pathogenicity even without the need of using living seedlings. So this system can be utilized to implement mass screening of tolerant material in a short amount of time. So in order to end the talk, I would like to summarize a few take-home messages. So first, the causal agent of the new and serious eucalyptus disease in North Sumatra was found to be a new species of Alcinoid, and we name it Alcinoid negatrix. Secondly, pathogenicity tests with Alcinoid negatrix result in symptoms identical to those under natural conditions. Current research is focused on determining the origin of the pathogens. A rapid screening technique is being established to effectively identify disease-tolerant planting stock. And finally, eucalyptus scab and shoot malformation represent an emerging threat to the plantation forestry industry in Southeast Asia, as well as other regions in the world where eucalyptus are planted. So I would like to thank all the funding body as well as all the collaborators to make this work happen. Thank you very much. Thank you for listening.
Thank you, Mr. Nampan, for the insightful presentation and on the eucalyptus, a new founding eucalyptus disease, Aeson necrotics. We keep your questions towards the end of the session, similar like the first session. So we go for the second session. Is it Dr. Rahayu ready? Mr. Here? All right, now. Okay, Dr. Insinyur Dr. IR Sri Rahayu, MP, is Associate Professor at the Laboratory of Forest Health and Protection, Department of Silviculture, Faculty of Forestry, Universitas Gadjah Mada. She has over 50 scientific papers and projects presented and published, and also international and national books published. He well recognized internationally, expert in forest pathology, especially on gall rust disease, on Falcatraria mulcona, and also on some disease of fast-growing species in the tropic, such as acacia and eucalyptus. She is the coordinator IUFRO Working Party on Tropical Forest Pests and Disease since 2010 and until 2024. She has good cooperation with many international research agencies such as USFS, with Hawaii, ACRAR Australia, GSPS Japan, AFAP, FISN, and the rest. And also many national research center and being an advisor to the government and private company Plantation in Indonesia. Without further delay, the floor is yours. Welcome, Dr. Sri Rahayu.
Okay. May I share with my own?
Yes, sure.
Yep. Just a minute. Okay. So.
Are you able?
Is it okay, my share?
Not share will be coming soon.
Okay.
Okay, then please zoom in. Set a slideshow, please.
Oh, okay.
Okay, it's perfect. Start from the first slide.
Yep. I'm sorry, it's very slow. Or may I close my, okay.
Okay.
Sorry, still in progress. Oh,
Yeah.
oh, sorry, it's very slow, my internet. So
Yeah. Take your time. Go to the first slide first before you zoom
yeah,
into the
yeah,
slideshow.
yeah.
Okay. Yeah. Okay. Take your time.
Just a minute. Hey.
Okay. Okay. Give your time. Edit.
Yeah.
All right. And please slide share.
Let's see. Hey.
Yeah. Otherwise, you can start to present from. All right.
Oh. Wow. Okay, just a minute. It is. Oh. Okay.
All right.
I'm sorry for the technical difficulties. Oh, okay. Very much for the time. So in this time, I would like to share our some of our finding of research about the characteristic of and pathogenicity of Ceratocystis fimbriata and Ceratocystis manginecans isolate collected from different sites in Indonesia. So many report and also many scientists already reported that Ceratocystis become a problem on our fast-growing plantation, including Acacia mangium and also eucalypt, and also some other species trees. So I will not discuss in this site because many experts more already discussed on that side. Yeah. So regarding to the problem, so we are from the Faculty of Forestry Gadjah Mada University, particularly in my laboratory, Laboratory of Forest Protection. We already collected about 130 isolate of Ceratocystis collected from many locations in Indonesia, including from Sumatra and Moluccas and Kalimantan, and also the latest from Java. We know that until now, Ceratocystis in Java Island, particularly on eucalypt and also on acacia, not yet reported. So my intention is to see whether the Ceratocystis isolate from the Java Island that never reported the pathogenicity, is it have a similar or maybe a different with other Ceratocystis from other location. So based on that, so we will to share about the morphological characteristic and pathogenicity of Ceratocystis fimbriata and Ceratocystis manginican collected from Java, Sumatra, and Kalimantan. Yeah, this is our collection. We have Ceratocystis from East Kalimantan here, and also from Sumatra. We collected from Subanjeriji, Palembang, and another isolate from Riau. But from Riau, we collected from Eucalypt Pelita. And also in Java Island, we have in Alas Ketu and Wonogiri, that is Central Java. We already collected here also, and we can see how is the characteristic and the pathogenicity compared to other location. This is for the standard method. Yeah, we trapping the perithecium using carrot, and after 3 until 4 days, we can get the harvesting the perithecium. And then also after that, we can isolate and use potato dextrosa agar in order to isolate some of the isolation that we use. Yeah, and then this is about 10 days with the similar age of isolate, and then we put take with 4, and then we inoculate, artificial inoculated on the seedling. That is the seedling egg is 3 months eggs, and then the seedling comes from F1 of Vipin provenant. Yeah, we have the Vipin provenant at F1 of Vipin provenant that already 6 years old in Wanagama. So we use the seed for the artificial inoculation. So after that, we can assess. This is just standard for ceratocystis inoculation. Yeah, we assess the disease severity, including the external discoloration and then also internal discoloration after the seedling already dry and dead. Yeah, this is the including the browner and trunk. And then now this is the result. We can see that the morphological characteristic, it is almost all the same. Yeah. So we cannot differentiate if we don't use the DNA marker. So we already identified that. Yeah, from Wonogiri we have Ceratocystis fimbriata and also Ceratocystis manginican. This is from Java Island, and then this is from Kalimantan, from East Kalimantan. We have also fimbriata and manginicans. Yeah, we check one by one isolate, and then from Sumatra we have from Subanjeriji, yes, Ceratocystis fimbriata, and this is from Riau. This is not from Acacia but from Eucalyptus pelita. And then based on the morphological characteristic, we calculate the size of perithecium neck length, and then osteolar hippa length, ascospora, conidia, and also chlamydospore. But we got actually all the accessories is almost the same, except we have the difference on Ceratocystis fimbriata from South Sumatra. That is, the conidia have the longest and the widest compared to other isolate. Yeah, notes that this is come from 100 sample. Yeah, we average from 100 sample of perithecia, hippa, and other. So this is the morphological of the ceratocystis, yeah, almost all the color are deep grayish olive, except the epe that is come from the Eucalyptus pelita, yeah, that is grayish olive. And also the growth of the colony, we can see that the EM4, that is Ceratocystis fimbriata from Sumatra, from Subanjariji, this is have fastest and largest of the colony area. And then we can see the pathogenicity. So based on the wilting of the percentage of wilting leaf, so actually three isolate cause the wilting in the same days, yeah. But another, this is the Ceratocystis manginecan from Java, and then also Ceratocystis fimbriata from Java, and yellow S fimbriata from Kalimantan. The three of isolate, this is have a more longer time, and then this is in 30 days. Still many seedling don't show the wilting, yeah. And this is the length of internal discoloration. We can see that actually Ceratocystis fimbriata and manginecan and also other. This is from Sumatra and this is from Java. The lowest length is manginecan from Kalimantan. That also one is from fimbriata from Java. And then this is the seedling mortality. Finally, four of the isolate, yeah, can make die of all the seedling in the 15 days, but two of isolate, including two isolate from Java Island, yes, Ceratocystis manginecan and so also Ceratocystis fimbriata, until 15 days, yeah, it still remain some seedling that not die. Yeah. So based on the molecular phylogenetic of the, this is actually we have SEC, yeah, already presented SEC, yeah, SEC Indonesian isolate of Ceratocystis fungi based on ITS DNA sequence constructed using Mega7 software, yeah, and the analysis involved 15 nucleotide sequence of Ceratocystis from several countries here. So we can see that we have two clusters. The one cluster is cluster of Ceratocystis manginecan with yellow color. Yeah, we can see that this in the first cluster, this is fixed. Yeah, Ceratocystis manginecan in the one cluster, and however in the another cluster we can cluster Ceratocystis fimbriata. We can see that in this here it is differentiated in many not in one cluster, yeah, but this is variety of Ceratocystis fimbriata. So this is still in doubt, yeah. Maybe that we know that Ceratocystis fimbriata still a complex of species, yeah. So maybe the more in-depth discussion about this cluster, because you can see that the Ceratocystis fimbriata that according to the pathogenicity, AM7, this is have a very high pathogenicity, yeah, but this is in one cluster with and one that this is low pathogenicity, yeah. This is based on the molecular phylogenetic. So I think more discussion have to do, and I know that maybe after the noon session, afternoon session, we have Irene Bernas she is very very good, and we know that many of research on ceratocystis have been conducted by Irene. She is a very expert in ceratocystis. So maybe after that, the discussion in depth will be presented on Irene Bernas, and we can see that the ceratocystis fimbriata, it actually still have many questions. Is it ceratocystis fimbriata or ceratocystis manginican? Because based on the molecular phylogenetic, we can differentiate, but for the pathogenicity, it is still in very variety. But in ceratocystis manginican, we can have fixed, the same species, and also have the same pathogenicity. Okay, finally, I would like to thank to Asia and also PT Musi Hutan Persada and PT Fajar Surya Swadaya East Kalimantan, who have support and also facility in the field during we collected the isolate and also conducted the artificial inoculation. Thank you.
Thank you, Dr. Rayu. You managed to keep your time. Thank you very much on that. And then we'll keep your questions later towards the end of the session. Okay, is it Mr. Su Gigi is ready?
Yes,
okay,
I'm ready.
You are ready. Okay, J.J. Sun is PhD candidate at the Nanjing Forestry University from 2019 to 2023. But currently you are at the University of British Columbia, Canada for 2 years attachment. And his doctoral research is mainly focused on the distribution and productivity of the main kinds of industry, forest plantation at the global scales under the context of climate change. Okay, for the title, Potential Habitat and Productivity Loss of Populus deltoides in Terrestrial Forest Plantation due to Global Warming. Without further delay, the floor is yours, Mr. Sun Gigi. Okay, you are ready. Are you planning to have your own slide?
I will try to. Sorry, wait a moment.
Okay,
so
you are ready.
You'll see my screen.
Yeah,
Okay.
it's clear, so you are ready.
Good morning, everyone. I'm a third-year PhD candidate in Nanjing Forestry University, and now I'm in UBC, Canada. Today, I'm going to introduce my previous study, which titled Potential Habitat and Productivity Loss of Populus deltoides Industrial Forest Plantation due to the global climate change. As we all know, plantations are usually established by a single species, and it makes up only 3% of the world forest area, but it supplies almost 40% of the world's timber. over the past decade, the rate of restoration of population plantation around the world has been slowed down. But the global timber demand is still growing in the future. And on the one hand, climate change will impact the productivity of the eucalyptus plantations. This study was based on ecosystem process model, and it showed the eucalyptus plantation located in Brazil are expected to experience increase of the productivity in the future climate change scenario. And on the other hand, climate change will also impact the suitable area of the forest or plantations. There are many studies showed the future habitat of some forest will decrease by the global climate change. We can see some wetland species and some normal tree species. Our study object is the Populus deltoides plantation. This plantation is very widely used for building forest plantation around the world, and the species is this species is native to North America and introduced to China, Europe, and many other countries. Under the global climate change, the productivity and suitable area of Populus deltoides plantation were facing large uncertainties. However, we still know little about this problem at global scale. Our scientific issue, first one, will the climate change have a positive or negative impact on the suitability and the total productivity of this plantation at global scale? Second one, where will the habitat and the productivity of this plantation increase, or where will loss in the future? And the last one, to adapt to the global climate change, how to adjust the afforestation plan and site selection for this plantation in the future at the context of global climate change. And now I will talk about the methodology. In recent year, using ecological niche modeling to predict the suitable area of forest has been very common and very effective way. The Maxent model was developed by the Princeton University, and it can successfully predict with more than 5 samples. This model was the most often used model due to its stability. We consider the 19 climatic factors from wood climate and 17 soil factors from the HWSD database and the global groundwater data from NASA in our SDM model building process. We selected 2 climate change scenario, which called the shared society economy pathway, SSP 126 and the SSP 585 in our model. Totally 2,600 locations were selected from the CNKI, GBIF, and the Web of Science. We also get our productivity data from the published articles. We surveyed the title, abstract, and conclusion of each download paper to identify the study that statistic the following criticals. More than 44 publications were considered in our study. Firstly, we do the homogeneity test of variance and the normalized test in R. Then we used the basic trendline package in R to test the relationship of productivity and environmental suitability to this popular Steatoides plantation. Here, I will talk about our results. We can see the AUCs in the current and future is close to 0.9. This means our model performed well. And we can also see the MTCQ, PDM, and the PDQ were the most important climate factors that determine the habitat suitability of this plantation. And we can also see the topsoil gravel content is the most important soil factor. Our model can also model this habitat suitability of this plantation response to the major climatic factor in the current climate condition, and the predicted high suitable area were mainly distributed in the United States, Europe, and China, and some area of the Australia. The predicted habitat suitability showed a significant positive relationship with the the productivity of this plantation showed a positive significant relationship with environmental suitability, and we can see the distribution pattern with high, median, and low production area of this plantation at global scale. When we combine with the 2 selected climate change scenario, SSP 126 and SSP 585, we further projected a potential loss of 2.5 to 3.3% in the global total productivity. from year 2041 to 2080. In addition to the global scale predictions, our results also recommended some adaptive management plans, such as for the areas with predicted increase in productivity in the future. Some enhanced afforestation is recommended, and while for those areas with projected decline in productivity in the future, some positive management measures like more irrigation and thinning were recommended. Collectively, this study presents a novel approach for predicting the forest productivity of populous deltoids in the context of global climate change, while providing a significant scientific basis for adaptive management strategies for these species to mitigate the impact of climate change. This study was finished in the May of this year, and it has been published on the Forest Ecology and Management. If you wanted to see more details, welcome to read and cite our study. Currently, I'm using the remote sensing technique combined with the random forest algorithm and species distribution model to simulate the future production of the eucalyptus plantation at global scale under the context of global climate change. And we have finished this. We also finished this study and ready for submission. Now I plan to finish my PhD project next year, and I'm looking for a postdoc position. Or if any professor who is interested in my research, please feel free to email me. That's all. Thank you. I finished my presentation. Can you hear me?
Elena, I think you're muted. We can't hear anything.
Hello, moderator. Hello, everyone. Can you hear me?
We can hear you, Mahdi.
Yeah.
We don't hear Kalina.
Yeah.
So can I proceed my presentation?
It's fine. I think
Yes.
everybody.
Please go ahead.
Okay, all right. Okay, thank you. Good afternoon, everybody. So thank you, moderator, and thank you to the organizing committee of IUFRO 2021 for giving me opportunity to present my paper on verification tolerance by Ceratocystis manginecans in clone of Acacia mengium selected pedigree progeny trial in Sabah. So my outline as follow: introduction, background to screening with two important points here: inoculum and inoculation protocol, and family screening. Where is the selected individual putative tolerant for verification confirmed, and then followed by clonal verification trial, and where to from here. So for introduction, Acacia mengium first introduced into Sabah in 1966 and planted commercially in Malaysia, approximately peak at 250,000 hectares in Malaysia and more over 2 million hectares through Southeast Asia. Ceratocystis cankerwill disease formally acknowledged in SSB around after Ceratocystis manginecans found to be causal agent of mass die-off in Acacia mengium. This is after Hello, any problem? Everyone? So why my slide not?
They're hearing you.
Mahdi, your slides are not moving, so you need to go into slide view.
Orang yang share screen kan, bukan kau kan? Iya, orang yang share screen. Siapa yang share screen? Kau kan orang.
I'm share the screen. I'm control the screen.
Kemudian kasih kontrol dia nak jalan.
Sorry, we start again, yeah. I stop share first. Hello. I start share the screen.
Yeah, Mr. Mahathir, I think you can share again and then test first. Your slide can be moved or not. Can you move to the next slide?
Presentation.
So the slide move already. So
the slide move. Yeah.
Mr. Mahathir, can you go to the slide view or slideshow? Click over there.
Yeah.
Slide view already.
No, because of the network is not good, but Mahathir,
yeah. We try the slide control from your side. Maybe can work.
Is it possible, Secretariat, to assist Mr. Mahathir to share screen? Means you are the one who's. Oh, Mr. Mahathir, probably you can switch off your video and then try to next slide and next slide.
Yeah.
Now, to be the PowerPoint version would match with the gadget laptop.
Secretariat, do you have any?
Hello?
Yeah,
Can you hear me?
Secretariat. Yeah,
Mr. Mahathir would need
Yes.
you to stop sharing your slide so that we
All
can
right.
share. Okay, thank you.
All right. Yeah.
Share screen.
So okay, I start from the beginning, eh?
All right.
So thank you to that organizing committee of IUFRO 2021 for giving me an opportunity to present my paper today on verification tolerant to infection by Ceratocystis manganecans in clones of Acacia mengium selected from pedigree progeny trial in Sabah. So next, please. Next, please. So my presentation outline as follow: introduction, background to screening program with two important points here, which is inoculum and inoculation protocol, and family screening, and followed by clonal verification trial, and the last where to from here. Next, please. So for introduction, Acacia mengium first introduced in Sabah in 1966 and planted commercially approximately peak at 250,000 in Malaysia and over 2 million hectares through Southeast Asia. Ceratocystis cankerwheel disease formally acknowledged in SSB around after Ceratocystis manganecans found to be causal agent of mass die-off in Acacia mengium. So please click the mass die-off in Acacia mengium. We have a happening slide. This is after field survey through transect line survey we do across Acacia mengium plantation in SSB, and the results show us the declining of MAI started at 4 years because of Ceratocystis manganecans and associated with animal damage also. So next, please. So in 2009, SSB has been planted alternative species trial with 15 different species, including local species, to identify other potential species can growing well in our local condition. So from this trial, 2 species showing the potential, which is Falcateria molucana and Eucalyptus pelita. So please click Borneo Forestry Cooperative to back to introduction slide, please. On the Borneo Forestry Cooperative logo on the top. Yes, please. Okay, so planting Acacia mengium ceased in SSB in 2012 and now replaced with Falcateria molucana and Eucalyptus pellita. However, arguably a place still for Mengium because of the adaptability, productivity, and also for species rotation in the future if needed. So that's why identifying disease tolerant Acacia mengium will provide substantial opportunities for plantation establishment and sustainability in the future. So next, please. So this is our simple screening strategy. So the aim of this study was to verify tolerance to Ceratocystis menginacum in over 100 putatively tolerant Acacia mengium clones selected from family screening. So both family screening and clonal screening, we do inoculation and selection after inoculation and bring them into clonal verification stage to reconfirm. But before we do this, we needed to find, authenticate, and test virulence of an isolate to use for our screening program. So next, please. So this is my colleague Rizwan looking the culture under microscope. So culture will isolate from symptomatic trees displaying strong typical symptom of canker and wilt. So sporulation was induced using moist chamber and carrot bedding on wood sample, and single ascospore measures were transferred into ontomel extract agar and supplemented with streptomycin sulfate. So pathogenicity of each culture was tested in preliminary trial, and single isolate was then used in trial. And from this result of preliminary trial, 4 weeks after inoculation, we measured the lesion length, and then the results show us Acacia mengium is the most susceptible Acacia species to Ceratocystis menginacum, while Alicoliformis and Carsicarpa is most tolerant. And this study also confirm that Ceratocystis fimbriata from sweet potato does not in fact propagated on these 3 Acacia species. So next, please. So I think this slide very important point I want to share, that in both family screening and clonal verification trial, but this photo taken from family screening, that the tests were conducted based on understanding that test trees required a well-developed vascular system to screen a vascular wilt pathogen. So you can see in this screen, the trees well established in the field, 1 year old, and this case, 1 year old, and same approach that we do in the clonal screening, in CSO, but the age were 3 years old. Okay, next, please. So this slide showing from the right side, showing the aerial view of our family screening. So you can see how aggressive our inoculum into the Acacia mengium species. So after 1 year, you can see only few trees left, only few trees present putatively tolerant, and this will be captured and put in our verification. And photograph on the left side, my colleague standing next to the big trees here, big diameter, because this is the trees that we inoculate, 6 years after inoculation. So, okay, next. So you can see the locality of these big trees 6 years after inoculation is come from this locality, actually. This is what I want to emphasize. So next, please. So similar in our clonal seed orchard. So based on the dramatic death of most of the clone inoculated in clonal seed orchard, it was decided that all trees in orchard need now be inoculated. So this followed the argument that there is some evidence for resistance being present, and the fact that more statistical significant test is now required to evaluate the result. So this photo shows that Acacia mengium is in clonal seed orchard after inoculation, showing some trees have survived. So a decision also made to inoculate the trees but not strip the bark from them to measure lesion length. This was to ensure that the bark stripping is not causing a new infection, and this skewing the data. So next, please. So this slide just to share visiting family screening on one of our field day, to sharing the update, the progress of the screening to our Borneo Forestry Cooperative members, and also from the delegates from outsider to share information and so on. So next, please. So okay, based on a selection for clonal verification. So this is our core presentation today. So in family screening stage, a large number of family Acacia mengium were inoculated, which is 100 different families. So based on the result, there was no clear evidence of resistance in family level, but individual tolerant trees within family being present. So over 200 individuals were selected, and this provides sufficient confidence to believe that it will be possible to identify trees with high level of tolerance, and trees could then be used in process towards to develop tolerant clone in breeding assets such as clonal seed orchard and control pollination or CP breeding garden. Potentially in the future also to produce hybrid clone with high level of resistance when we use this high tolerant Acacia mengium to cross with Acacia oligoliformis. The selected individual trees were captured, then planted again in verification trial with this, you can see in the screen, 6 different trials. Okay, next. So this is the table, a summary of our screening verification trial. So we have 6 different trials, different number of treatments. So you can see column number of clones. So we started from 15 clone and until the letter of trial 55. So this is the interesting information. If you can see both column between number of clone and number of family. For example, in clonal screening number 1, we have 15 clone, but from also 15 family. That means this information tell us only one positively tolerant clone from each family. Similar to interpret for the other trial below. For example, the later trial, clonal means clonal trial 6. So 55 clones from 37 families. That means we have some family present more than one positively tolerant. Okay, for design trial, so you can see single tree plot in the first place, and then we change a little bit to 3 single line tree plot. And then also the stocking, the stem per hectare. Initially, in first place, we use 444 stem per hectare, which is so dense, 1.5 meter by 1.5 meter, because we thought that last time this trial we will be inoculated less than 12 months. But after we understanding that the vascular system required to see the response of inoculation, so that's why we changed the design to normal spacing, which is 3 meter by 3 meters, so we can get 11 stem per hectare. In terms of inoculum, so you can see T75 actually our local ID actually similar with Mike Winfield ID CMW that we use in our pre-inoculation trial just now. But you can see the letter after the inoculum ID C, D, D, F, F, G. So means that we skip juvenile the culture to make sure we can maintain aggressiveness of culture before we use for the screening. Okay, so for criteria assessment, after 12 months after inoculation, we decide to use these 5 traits: survival, crown hull, and we add 3 additional traits: gamosis, sunken bark, and boro. If we can see the correlation. Okay, next please. So in terms of heritability, so you can see here from the result. So improvement in heritability estimate with successive trial. So from 0.07 to, for example, survival 0.07 to 0.2. So improving, this could be either due to general trend towards high treatment numbers in the later trial, or improvement in phenotyping over time. So, but heritability was pretty low for most. High genetic correlation for survival, crown health, and sunken bark indicates high repeatability for the screening for this trait when the same clone were evaluated in different screening trial, because we have clone linkages between SB6 trial. Okay, although this is no value for sunken bark with extremely low heritability, but actually when we measure the trees at 1 year after inoculation, so difficult to see the bark already because the trees already dry and broken, and only we can see borer. Okay, next please. For correlation, so logically, survival and crown health, of course, positive correlation. So this data also show the positive correlation 87%. But we go further down in terms of borer. So borer incidence indicate low survival and also low crown health. So you can see negative correlation there, 46 for survival and 47 for crown health. Okay, when we go further down, gamosis incidence indicate high gamosis indicates that high crown health and increased survival to 77 and less borer. Okay, and maybe gamosis production on this time, 12 months after inoculation, is positive response to infection. But sunken is not heritable and not associated to other traits. Okay, next please. So, okay, this study showed that survival and persistence of healthy crown after inoculation was heritable after inoculation. So, therefore, provide potential to bring together relatively rare genes in breeding assets such as clonal seed orchard and also control pollination garden, breeding garden. Rare means here is because, as I mentioned in the early slide, we have 2,000 individuals were inoculated, and then after 1 year after inoculation, only 9% from them present putatively tolerant. And from this 9%, finally in verification trial, we only get 2%. So post hoc analysis in this trial can now be used to cross-reference breeding value for this high heritability, which is survival or crown health with this selection. So you can see on the screen, this photo shows some of surviving trees that we have subsequently captured and established in our OP seed orchard and CP breeding garden. So this underground selection were made at a completion of each verification trial amounting for a total of 50 clones, which is equivalent to 2%, 50 clones that display strong evidence of tolerance. So next slide, please. So this 50 clone that we select have been recaptured and established in this seed orchard and breeding garden. So you can see on the screen on the left photograph on the left side, our colonial seed orchard at 1.5 years old, and then on the right photograph on the right side in your screen, showing our CP breeding garden, where we try to do a hybridization, control pollination with this high tolerance medium to this Oligoliformis to try to produce high resistant level acacia hybrid in the future. Okay, next, please. So from this study, I made 5 actually important conclusion to for this study. So first, acacia menjum has all but been erased from this industry replantation landscape in Sabah due to ceratocystis canker wilt disease. And screening program for this screening program, such as this one, provide opportunity to identify tolerant clone for future utilization of industrial tree plantation. And the next, this study showed that survival and persistence of healthy crown after inoculation was heritable and repeatable. So next, please. And the potential to bring together relatively rare genes for tolerance from the screening program, SSB and BFC has made possible to capture of 50 clones that have been tested and verified tolerant to ceratocystis canker wilt disease. This can now be included in clonal and breeding trial. And the last for my conclusion, this study was based on the understanding on preliminary trial with ceratocystis mengenekon that test we required a well-developed vascular system to screen for tolerance to this vascular wilt pathogen. This is the first time that study of this magnitude and conducted on established trees has ever been undertaken. Okay, next, please. So acknowledgement, because this study I not doing by myself alone. I have strong people behind me, so I want to acknowledge this group of important person, management team, and my colleague at Sabah Sopot Berhad, the company member of Borneo Forestry Cooperative, always support me, share information. And the third one, BFC support scientist. Of course, this is the core behind me to always support me every time to make sure this study get the objective. So like Mike Wingfield, Paul Wolberton, Jeremy Browner, and David Bowden. And the last but not least, University of Pretoria, Fabi.
Thank you.
Okay, next, please. Next, please. We still have, next, please.
We still have, right.
Field photo during assessment, inoculation assessment with this group. So next, please. Next, please. Okay, so this is my last slide, I think, but look how beautiful this species, yeah, young species of Acacia menjum plantation. But we must stop because of this ceratocystis mengenekon. But after we have the result from the clonal verification, and now I can say that I strongly believe this species will come back to change back our landscape of tree plantation in near future. With that, thank you so much.
Thank you, Mr. Mahathir. Very motivational, and then have put a very highful hope to the Acacia Museum to return back to Malaysia, definitely at the Sabah Wood. He's a very experienced 16 years tree breeder at the research development departments of at Sabah Softwood Berhad. Okay, now is the time for the Q&A. You can see from the Q&A section there is a questions given for Mr. Nampai. Okay, please.
Okay. Start the questions from Istiana Priadini. If by any chance the pathogen cannot grow on agar mediums, how we can identify the unknown pathogen from these plants? And how can we do the pathogenicity test? Okay, so Alcinomegatrix, yes, it's difficult to isolate due to a couple of reasons. First, it's a very slow growing fungus, notoriously slow growing, and it can it can be outcompeted by other fast-growing contaminants or secondary things growing on the same disease symptoms. Like I show on the presentation, like we got like a huge diversity of other fungi and yeast as well, but it doesn't mean this cannot be isolated. Alcyon negatrix is not biotroph. That means it can grow on culture. So yes, it's difficult to isolate, but you still be able to isolate, but you just need to isolate the right way. It took a long time to be able to isolate because it's the first time we're working with such group of fungi, and now we have the experience and we have the protocol described in the paper how to isolate with media as well, like special type media. And also we are in the process of developing other tools to help with the identification identification or detection of the disease, like for instance LAMP PCR, so you don't have to do the isolations. You just need to extract the DNA from disease material, and then from that you can be able to tell if your plant got infected by Alcyon negatrix or not. And about the pathogenicity test, the thing about Alcyon negatrix is they are sterile in culture. That means they don't sporulate on culture. So the way we do in pathogenicity trial is with mycelial fragments. Even with mycelial fragments, we still can be able to reproduce the disease that similar to the natural infections. And when we check with under the scanning electron microscope, then we can see the sporulation on the plant material. So yeah. I hope that answers the question.
Okay. Thank you, Mr. Namfan, for the clarification. Okay, there is one question for Mr. Mahathir. Would you mind to answer that at the chat room, Mr. Mahathir? We will be seeing the Acacia management plantation in Sabah. Sorry, you you are muted. We can hear you, Mr. Mahathir.
Okay, sorry. Okay, I cannot see which one ask the question.
From Doringo.
Oh, from Dorengo. Okay,
yeah.
Okay. Hi, Dr. Doren. So yeah. Wait.
Okay.
Dorengo. Wait.
Okay.
Oh, Mahathir will be seeing a major plantation.
Yeah.
Okay. So as I mentioned in our slide, in our CSO planted 1.5 years ago. So expecting will produce seeds. I think 1 more year. But as I mentioned in my slide, these seeds from clonal seed orchard yet to be demonstrated that the tolerance level can pass onto the progeny. So, but we need to demonstrate. But in terms of the availability of seeds from the high tolerance clone, so will available 1 more year.
Okay, thank you.
Okay,
there is another questions in the Q&A section. Can you click that? And then from Sri Sunarti.
From Sri Sunarti.
Okay.
Could you? Okay. First thing, I'm not doctor. Yeah. So just Mahathir. So could you please inform to us a little bit more about generation of breeding Menjum genetic material to be tested? Oh, okay. So as I mentioned in slide, this is in our family screening containing 100 selected family, actually selected from the first generation progeny trial, which is introduced in 1990s through ACR project last time. So this progeny consider a broad genetic population because we also include our local selection and brought, and we consider second generation progeny. Yeah, but mix, actually mix 1.5 mix, but selected families. Yeah.
Okay,
I
thank
think
you.
an answer. Yep.
Okay, Mr. Nampan, there is one suggestion from Mike. There is at the chat room, thinking about the NAMS presentations. Can you find that?
Okay.
Okay,
So it's
please.
like a comment from Prof. Mike Winfield.
Mm-mm.
So we know this serious disease only from North Sumatra. So yeah, from our research. So currently it's only in the North Sumatra regions, but it will spread and it seems likely to spread. So like my suggestion, we could encourage the forestry, the regions to be aware of this disease and its symptoms and look out for it. And we will be able, we will be willing to help with the identifications if yeah, with all the process if needed. Yeah, because it's the important disease need to be looked out for. Yeah.
Okay. I think Mike's you happy with the answer. Okay, there is one question from Eco, but it can probably match with Dr. Rahayu, which is a better strategy to develop acacia resistant or tolerant. Acacia, please.
Okay. This is the question and answer from Mr. Dr. Wong. Is it? Hello?
Yeah. Which doctor?
Yeah. This is the question is from your presentation slide. Acacia sp and eucalyptus sp will be facing the same problem, which attacked by the ceratocystis. If possible, to plant the eucalypt to replace the acacia plantation, which already attacked by ceratocystis. I'm sorry that because now we still also anticipate that maybe the next time eucalypt also will be devastated by the ceratocystis. So we still research earlier, yeah. So this is for anticipate if finally we got also problem with eucalypt. So we have a base study and we have also information maybe with the clone will be tolerant, because this is the problem on our plantation. Yeah, we know that plantation with fast-growing species will always problem, yeah, with pest and disease regarding to the ecosystem, yeah, that is monoculture and like that. So we have to aware that in the future we will have always the problem. So don't do the test or don't do the research after the problem already faced. Yeah, we have to anticipate before. We have to have maybe many researchers also already mentioned that we have to monitor, and then with the monitor we can predict what's the next problem. So we have to anticipate, prepare earlier, yeah. But I don't know which you. Of course, I think eucalypt will also have is facing the problem also, yeah. But I don't know when. So because many publication now still dominated on Acacia magnum, yeah, and also maybe Acacia crassicarpa, and the next time will be problem, yeah, because we also planted in many location in gambut, yeah. That is also the ecosystem is very very different also, yeah. I think I'm sorry, that is my answer. Thank you.
Okay, thank you. Mr. Mahathir, there is another question from Dr. Darren Go. Is any plan to propagate the 50 clones selected for the tolerance? You are muted again, Dr. Mahathir.
Yeah, for your information, this 50 selected clone already introduced in tissue culture lab. So if this in tissue culture available, so easy for us to if we have how to call a further utilization. If this all 50 clones already in tissue culture, but now in tissue culture already, we have in CSO available, and also we start propagating through a tissue culture.
That's a great news. We do hope it can be materialized soon. So it's wonderful. Okay, Dr. Iko Hardianto, your questions, is it aimed for who to answer your question, please, Dr. Iko? Probably you can ask. You're muted, Dr. Iko. Yes, please, Dr.
Okay,
Iko.
sorry.
Yeah.
Sorry. My question is directed to the Mr. Where's the last speaker from Sabah, Softwood? Oh, to Mr. Mahathir. Sorry.
Yeah.
Okay.
Yeah, Dr. Eko, please.
Okay.
Yeah, my question is whether the Sabah Softwood will develop the Acacia mangium pure species that resistant to the ceratocystis, or the hybrid Mangium and Aeroplipormis, considering that Mangium is difficult to clone and also has a problem with aging, you know. So the road for vegetative propagation is quite unfeasible for Mangium. So probably we will use a seed, but the question is whether seed is quite inherit the genetic resistance from the parents. That's my question.
Yeah, of course.
You get my point?
Yeah. Okay, I got your question. So my answer like this. For development of CSO to produce seeds and development on CP breeding garden, yes, we propagate easily like grafting or cutting. But of course, we realize that this is aging problem to propagate. So that's why we go for hybridization. So to produce, everybody I think well known that Acacia hybrid is easy to propagate. So that's why we will go to produce Acacia hybrid soon as possible, and then from that we can propagate through cutting. Yeah.
Okay, thank you.
Yeah.
Okay, thank you. But in the Q&A room, you can still have one question from Sri Sunarti about the progress of hybridization. I think that question goes to Mr. Nampan, right? It's a continuous question from the first one. Yeah. Everyone can. Yeah, please.
I think if the audience asks about the hybridization, might be related to my presentation.
Yeah, please proceed.
Yeah, so the status of hybridization, yeah, we start actually last 3 years from our CP garden. So now in how to call in hybrid selection block. So now we start selecting the putative Acacia hybrid. And then when we captured from this selected putative hybrid clone, and then we must through the process, the same process with the what I present just now. We need through the process, all the process with verification and also how to call the clonal verification, of course, for sure. Because not all Acacia hybrid that we produce is we confident that highly tolerant. So that's why we need to screen also to make sure that the selected clone with high level of resistance that propagated for commercial deployment. Okay, I think this is from my view.
Okay, thank you, Mr. Mahathir. Mr. Arif, do you have questions?
Okay, yeah, I have question for Mr. Mahathir.
Okay, please.
Thank you very much for your presentation, Mr. Mahathir. That you just mentioned that your genetic material for your research is coming from the first generation of breeding. That's right?
Sorry, from
the first generation Yeah.
Yeah.
Do you have opinion? Is there any relationship between the largest genetic variation? So we will have some potential to get more tolerance genotype. Yes, all right. The larger genetic variation. The relationship between.
I not really, but as I presented just now, in our screening program with 100 different family brought genetic population, but we cannot see the tolerance in family level, but only individual within family that can show the present the putatively tolerant. So that is I can say just for the time being, based on my genetic population that we put in our screening program. Yeah.
Okay, thank you.
Yeah.
Thank you.
Yeah.
I think the other question was not appear anymore. Okay, clear. And then in the chat room also, there is no new, but there is some noted given by Mr. Paul. BFC members successfully transition of eucalyptus pelita in Sabah some time ago, along with vigorous tree improvement that includes disease screenings. Thank you very much to all the speakers, Dr. Rahayu, Mr. Mahathir, Nampan, dan juga Sujinji. Thank you for your very fruitful discussion, information, and very informative, insightful presentation. And we wish to meet you again next time. For the next section, we start at 2 p.m. And then we have Simon, Kira Lane, as well as Havenga for their very interesting presentation. We meet again at 2 p.m. session, and then we end now our session today. Thank you very much.
Thank you again, Dr. Khalida. But we definitely will see again in the afternoon to continue the risk management sessions. A special thanks and congratulations again to all speakers for this morning sessions. Dr. Anto Rimbawanto, Dr. Mike Wingfield, thank you. Dr. Stuart Fraser, Mr. Nampan, Dr. Sri Rahayu, Sanjiji, Mr. Mahathir Lamunu, for a very solid presentations and definitely beneficial for a more robust forest plantations management, especially in the area of pests and disease management. Ladies and gentlemen, we will be having our lunch break. We will see you again at 2 p.m. later. Thank you again. Distinguished participants, ladies and gentlemen, we will now resume our sessions on risk management. Again, I would like to invite Dr. Kalima to continue her role as the moderator for this session. Dr. Kalima, please.
Thank you, Ms. MC. And then today for the afternoon session, we have three presenters for the first one, which is Associate Professor Dr. Simon Lawson. He's ready in the screen. Okay. Simon is Associate Professor in Forest Health at the University of the Sunshine Coast and Director of the Biological Control of Eucalyptus Pest Alliance, BICP. Prior to joining USC, he was team leader for forest health research in the Queensland Department of Agriculture and Fisheries, DF. He has over 40 years experience in forest entomological research in Australia and internationally. His research focused in developing effective, sustainable management methods for insect pests of plantation forests. With an emphasis on biological control, he also has interest in forest biosecurity, population modeling, and international collaboration in forest health and biosecurity. Without further delay, the session is yours. Please, Dr. Simon.
Thank you, Dr. Kalina. May I share my screen?
Yes.
Okay, I hope everybody can see.
Yep, yeah.
Oh, okay, good. Okay, well, I'd like to firstly thank the organising committee for inviting me to present, or allowing me to present today. It's been a great session. I must say there's been some fantastic talks. I'm going to be talking about something a little bit wider than specific issues with acacia, but across forest health and biosecurity more generally in our region in Southeast Asia. So just to set the stage and see what we're facing in terms of threats from pests and diseases, this is an accumulation curve for invasions of eucalypt pests, actually from Australia to overseas. And as you can see on the x-axis, these insects have been moving around right from the middle 1800s right up until the present day. And on the y-axis is the cumulative number of overseas introductions of these Australian insect pests. So if you look closely, you can see really the accumulation curve divides up into two sections. One is this section from the 1900s around about to 1990, where there was basically a slow accumulation of new invaders. On average, one new introduction of an Australian eucalypt pest every 2 years, and then quite slow additional movements between countries. And then since the 1990s, we've seen an acceleration of this faster accumulation of new invaders, almost one new introduction every year in the last 20 or 30 years. And then with international trade, very rapid additional movements, what we call the bridgehead effect. So once a new pest is in one country, it's much more likely to move into more countries. So this is just setting the stage for what we're facing in terms of movements of international pests. And then also just setting the stage in terms of plantation forestry in Southeast Asia. We've heard a lot from everybody talking about this already this morning, so I won't go into too much detail, but we've really been talking about the bulk of the plantations are mostly centred around acacia and eucalypt species in this part of the world. And also we've heard quite a lot about the amount of investment that's been going on in development of improved germplasm for these species right from the start. Australia was involved in this, and now quite a lot of investment from private industry and other governments around the region in this research. And there's a real need that others have talked about for improved biosecurity to try and protect these investments from emerging pests and then better manage them, better management for them once they actually do arrive. From an Australian point of view, these plantings of acacia and eucalyptus throughout Southeast Asia and elsewhere in the world act as kind of sentinel plantings for us. They give us early warning of new pests and diseases that may be developing, and we've seen a few of these referred to already, particularly FAM's presentation. And a lot of other countries around the world are deliberately going and planting their tree species elsewhere to set up sentinel plantings. But for Australia, the industry has kind of been doing that for us for free. So I guess we should be very grateful for that. The other side of the same coin is that these plantings also can act as potential pest sources for Australia. So we have an interest, a vested interest also in looking at what's happening in acacia and eucalyptus plantings in our region. These are very approximate areas of plantations of eucalypt and acacias across the ASEAN, most of the ASEAN countries. And I guess really the take-home message here is, and other presenters have talked about this, is because of the disease issues with acacia, the plantation area of acacias has been decreasing. The red arrow next to the 3.6 million hectares across the region, and the increase, eucalyptus area has been increasing, now around 1.3 million hectares. And really those three countries in green at the top have quite mature industries, very different industries, but quite mature in the way they plant and in the industry development. Whereas the other three countries, Laos and Cambodia in particular, have very young and developing industries. Malaysia also, you could say, particularly in Sarawak and Sabah, as we heard this morning, with ambitious goals to increase plantation areas. So that sets the stage for what's been going on in terms of research in forest biosecurity in Southeast Asia. And there have been a couple of recent Australian Centre for International Agricultural Research projects in the region, that's ACO projects. First of these is one which I was involved in, which finished 2 years ago, which was biological control of galling insect pests of eucalypt plantations in the Mekong. And another one centred from the University of Tasmania around management strategies for acacia plantation diseases in Indonesia and Vietnam. And Dr. Antol provided some of the results from that work earlier this morning. These projects finished almost at exactly the same time, and in the project final reviews, the four partner countries really identified a need to enhance regional capacity in Southeast Asia to monitor pest threats and respond effectively. So this led to the development of an ACR small research scoping project, so scoping for a forest biosecurity network in Southeast Asia. And we've just finished that project just in the last year. And the outcomes of this project really centered around a number of different outcomes. The first really was around the need to determine what the current biosecurity capacity, knowledge and priorities for stakeholders in the region is. And I'll go into a little bit more detail of the results of this shortly. We also reviewed the regulations and laws and trade conventions and treaties for biosecurity in the region, and we've just submitted a paper for publication potentially in the International Forestry Review. A really important thing to do also was to review the past and current investment in biosecurity in the region. So for future work, we don't want to be wastefully repeating work that's previously been done in biosecurity in the region, particularly around things like diagnostics and surveillance, because many of these skills and methodologies are transferable to forestry. And lastly, we began initiating this network across the region, which we hope will deliver better coordinated responses and an enhanced capability in the region. And one of the main things that we see a need for is particularly bridging the gap between forestry and agricultural biosecurity agencies. They often sit in different departments. They may be in the same ministry, but they're in different departments. And historically, the connections have not been good, at least in some countries, including Australia, until comparatively recently, between forestry and agricultural biosecurity agencies. So better collaboration we see is very important. So just going through some of the outcomes in terms of determining capacity gaps and training and knowledge needs. We originally were going to have a workshop at the beginning of last year, in March last year, and then COVID intervened. And so all of this sort of work moved online, which turns out to have been maybe a good thing. We've maybe been able to get better information online, using online methodology than we may have done in a more limited workshop. So for training and knowledge needs, we had a questionnaire which was on a Likert scale of 1 to 7. And you can see here, these are summaries of individual responses to questions around what training and knowledge needs do you have. And so the top one there is the management of risks of importing new quarantine pests. The second ranked was early warning systems for forest pests. Then diagnostics was also an important one. Creating forest pest lists. And then amongst the top listed ones was forest pest and disease surveillance, which also links to the early warning systems. And quite a lot was still really quite highly ranked when you think of the scale of 1 to 7. Quite a lot are in the range of 5 to 6 as well. So those generally fit pretty well with feedback that we'd had from the other previous projects. Another way of looking at this was to see what congruence there was in these training and knowledge needs actually across the countries. So the first summary was individual needs, and then this was looking at how much agreement there was across the countries. And there was pretty good agreement between the individual needs and the country needs. So diagnostics and early warning systems were the top 2 ranked. Training and knowledge needs. Then treatments of commodities was one which perhaps wasn't as highly rated individually. And then management of risks of importing new quarantine pests, pest risk analysis, and then forest pest and disease surveillance, again, was quite a highly rated one. We also looked at skills needs, and these were, again, we looked at individual responses first. And so we have the top ranked response was influencing government regulations and policies was a skill need. Negotiating biosecurity issues with trading partners in other countries was another one. And then a whole range that were very, very close, including discussing forest biosecurity issues with your colleagues in other countries. And again, we looked at this for agreement across countries. There's not quite a complete agreement across countries with these as they were with the knowledge needs. But the top ranked priority skill need was negotiating biosecurity issues with trading partners in other countries, and then finding technical information when you need it, influencing your organisation's strategy and operations, and then discussing forest biosecurity issues with various stakeholders was an important skill need. As part of this process, we also set up discussion groups on WhatsApp, and then also via an online question with free-form answers, and this was really valuable. And there were was quite a lot of agreement amongst our participants on what were the key gaps and what were the things that really limited their ability to deliver on biosecurity activities. So limited funding, limited training opportunities, poorly equipped laboratory facilities, and lack of skilled and trained personnel were some of those. And all those things limited people's ability to conduct their jobs and limited the more general biosecurity capacity of each of the countries. And whilst governments in the region do provide training opportunities in a range of areas, including policy, diagnostics, surveillance, and mapping, these opportunities don't occur that frequently and often limited in the number of places available. And a lot of respondents also indicated they wanted more training in things like diagnostics, surveillance, and new technologies. So again, this was a bit of a silver lining of moving these discussions online, because I think we got a broader range of opinions, and we probably got some more frank opinions from people who are able to anonymously express their views. So the outcome here now is that there have been 2 new ACR projects developed, and they've just been signed off by ACR in the last few weeks and days. And these 2 projects are really around the science and policy that's needed to support better forest biosecurity in Southeast Asia. So Anto, this morning, Dr. Anto introduced you briefly to the first of the projects, which is managing risk in Southeast Asian forest biosecurity, to be run out of the University of Tasmania with partners in Vietnam and Indonesia. And the main things that are happening in that project are screening for disease and pest tolerance, cost-effective drone technologies for early pest detection, and modelling climate change impact on biosecurity risks. The other project is one that's focused on building an effective forest biosecurity network in Southeast Asia, and that's run out of the University of the Sunshine Coast. And we have 6 partners in Cambodia, Indonesia, Laos, Malaysia, Thailand, and Vietnam. And our aim is really around to enhance the capacity generally in biosecurity, improve networking, get some better surveillance, diagnostics, and reporting, leading to overall better forest biosecurity in the region. So the first project with the disease screening and breeding, there's been a lot of discussion and just in the last session in particular, and all morning, about the importance of being able to provide better germplasm. So we've heard about great work that's been going on in the industry in Indonesia and also in Borneo through the cooperative there as well. So there'll be further work done on this in this ACR project. We've also seen yesterday presentation on using drone technology, in particular remote sensing, for early detection, and that'll be another big focus of this of one of the projects. And the other one, modelling climate change impact on biosecurity. So we all know the complexities of climate change, how there are sometimes winners and losers and positive and negative effects. And the project will outline sort of complexity that's in this diagram, with added complexity about invasion biology and the ways in which climate change may affect invasiveness of particular pests and diseases in Southeast Asia. The other project, which is the one that I will be working on mainly in our team, is the network, building a biosecurity network in Southeast Asia. And we've designed this in such a way that the network focus is going to be on high-risk site surveillance. And this is really a learning through doing exercise. So high-risk site surveillance is establishing trapping and general surveillance at high risk sites such as airports, seaports, container depots, these kind of sites where pests and diseases are most likely to enter a country and to establish. So we chose high-risk site surveillance as the centrepiece for the project because it really does assist with a lot of things and points out lots of things you need to do to have a good biosecurity system. So it really does assist with, you've got to know what pests you've got to know which ones you want to keep out. So, so it provides baseline data on what pests you already have, which gives you the information you need to make better decisions about what pests you don't want in your country, which leads further down to doing proper pest risk analysis to look at the pathways in which those pests and diseases might arrive. And then to identify and categorise the risk sites using pathway analysis and further risk identification. And then once you've done that, we've got those pest risk sites, and it's a matter of establishing and deploying these effective methodologies for early detection. And the photograph on the right shows a beetle trap, which is one of the techniques that we'll be using. High-risk site surveillance also assists with determining diagnostic capacity gaps. So if we're trapping lots of things, we need to be able to identify them. We'll soon determine how much diagnostic capacity there is to be able to do that and what the needs are in countries to get better diagnostics.
Dr. Simon, you have 1 minute left.
Okay, thank you. We're almost there. And then we also need to report what you find. So sharing our data and being able to act on our national and international reporting obligations. And also, if we do find something in a trap at a port, are there mechanisms in place within the country to mount an incursion response? How do we respond if a new pest or disease arrives? And then all of that really, we hope, flows through to better informing biosecurity planning and policy development. And that really requires collaboration and cooperation across the entire region. So we see the value of these projects will be the creation of an effective and sustainable forest biosecurity network for Southeast Asia. And it really is important to try and make sure that this is sustainable. And we want to do that by having a fairly large part of the project developed towards policy and future policy that values forest biosecurity, and so therefore direct investments in biosecurity into the future in the most cost-effective way within the context of each country. So our goal is to have our forests, or keep our forests healthy, and there's a lot of talk now in biosecurity around One Health. So One Health is integrating health of the environment, health of our animals, health of our plants, and human health into this thing called One Health. So we're part of that. And with that, I'll say thank you, terima kasih. And if you want to contact me, my email address is there. So I'll say thank you very much.
Thank you, Dr. Simon. Thank you very much. I think that Kira is ready with online. Okay, she completed her undergraduate Bachelor of Science Honors and Master degrees in Forestry and Agricultural Biotechnology Institute of from Faculty of Natural and Agricultural Science of University of Pretoria. Is currently a second year of PhD studying in genetic diversity in the fungal pathogen Ceratopsitis in commercial forestry plantation of Indonesia, South Africa, and Brazil. Okay, you are ready with your slide, Kira?
Yes, I can share them.
Okay, please. Can we have a look?
All right, can everyone see?
All right, yeah, it's great.
Okay, thank you so much for the wonderful introduction. And thank you so much for the opportunity to allow me to present some of the work that I did in my master's, which focused on 2 Urulaceae ambrosia beetle species, their new Fusarium species, and their potential impacts on commercial Acacia crassicarpa plantations in Indonesia. So firstly, ambrosia beetles are wood-boring insects, the life cycles of which are spent almost entirely within the xylem of suitable plant hosts. Here you can actually see a feld-infested Acacia crassicarpa tree that has a beetle gallery within it. They're able to survive in this nutritionally poor environment due to their obligate association with fungi. Here you can actually see a fungal garden growing within a beetle gallery. And here you can actually see an ambrosia beetle feasting on its fungal associate. They store and transport these fungal associates in specialized structures termed mycangia, which are indicated in yellow on the diagram to your left. These can be located on different parts of the beetle body, depending on the beetle species. So ambrosia beetles can be associated with a wide variety of fungi. However, those that are stored and transported in the mycangia are specific and termed ambrosia fungi. In the ecosystems of their tropical and subtropical native environments, most ambrosia beetles live in decaying wood and largely remain economically inconsequential. However, due to certain interactions, particularly their accidental introduction in non-native environments, several ambrosia beetle species have emerged as important pests, particularly in the tribe Alloperini. So the shift in the virulence of, I don't know if it's me that's the echo. So the shift in the virulence of these microbial associates from benign nutritional associates to phytopathogens is poorly understood. But it does seem that the increase in these ambrosia beetle-related diseases is due to activities that facilitate naive encounters between the beetle-fungus symbiosis and new plant hosts. More specifically, it appears that these increase in these diseases is due to the interaction between the beetle's microbial associates and plant hosts that lack co-evolved adaptation. Therefore, identifying the microbial associates of these ambrosia beetle pests and determining their diversity and pathogenic potential is really pivotal to understanding their disease biology. So as most of the important ambrosia beetle-related diseases have been studied in their non-native environments after they've been accidentally introduced and have caused significant economic damage, the diversity of these ambrosia beetle species, as well as their fungal associates, is most likely underrepresented. So this hypothesis was reiterated by a recent study which investigated species within the Eulacea fornicata species complex in their presumed native range of Taiwan. And in this study, they found that these beetle species were associated with a wide range of fungi and thus had a more promiscuous and more diverse association with their microbial associates than what was previously reported in their invaded environments. Therefore, investigating these emerging ambrosia beetle diseases within their native environments can hopefully provide us with a better management strategies on how to manage them and also hopefully reduce their potential impacts if they're reduced into other non-native environments. So for this study, we decided to investigate the gradual increase in ambrosia beetle infestations on Acacia crassicarpa plantations in Riau, Indonesia. Now, this increase in ambrosia beetle infestations was not only a cause for concern and needed to be investigated because we didn't know why there was increase in this ambrosia beetle infestations, and we didn't know where these beetles came from, but this disease paradigm actually also provided a really great opportunity to understand an emerging ambrosia beetle disease in its native environment and could hopefully allow us to give more insights into this ambrosia beetle symbiosis and the diversity of hopefully other ambrosia beetle related diseases. So briefly, what we did in this study was Acacia crassicarpa plantations that showed heavy ambrosia beetle infestations in Riau, Indonesia were identified. and 5 geographically separated compartments were selected as collection sites. From these sites, several heavily infested Acacia crassicarpa trees that were still living were felled at 50 centimeters above ground level and cut into 50-centimeter logs. These logs were then transported back to the lab, where the ambrosia beetle galleries, as well as the ambrosia beetles, were dissected. The fungi that were isolated in this study were either isolated directly from the surface of the beetle galleries or from the heads of female ambrosia beetle species. And this is because this is where the mycangia of these beetles was located. These beetles, as well as the fungal isolates, were then identified based on morphology and DNA sequence data. So what we found was, based on a maximum likelihood phylogenetic tree using the COX-1 gene region, we identified Eualaceae per brevis in green and Eualaceae similis in blue as the 2 most abundant ambrosia beetle species infesting these Acacia crassicarpa plantations in Riau, Indonesia. Upon closer inspection, we also identified several novel haplotypes of these ambrosia beetle species. These ambrosia beetle species can be distinguished based on their morphology, and they also seem to differ in their ecology, with Eualaceae per brevis seeming to be a primary borer able to infest seemingly healthy plant hosts, whereas Eualaceae similis appear to act more in line with typical ambrosia beetle behavior and could only infest plant hosts that were already colonized or compromised in some way. When investigating the fungal associates of these 2 ambrosia beetle species, we identified 3 new Fusarium species of Eualaceae per brevis, with Fusarium rheacanum being the most abundant, as indicated by the thick red arrow. The prevalences of these Fusarium species differ and are indicated by the thickness of their arrows. When looking at Eualaceae similis, we were able to identify 5 new Fusarium species, of which Fusarium 1 was the most abundant. As Eualaceae similis appears to be a secondary borer, we believe it has the ability to acquire associates of other closely related beetle species, and is most likely why it has a more promiscuous relationship with its fungal associates than Eualaceae per brevis, which is a primary borer. We were also able to identify another new Fusarium species, but we could not confirm its beetle vector, as this Fusarium species was isolated directly from the beetle galleries. All in all, we were able to identify 7 new Fusarium species that were associated with these 2 ambrosia beetle species. These new Fusarium species have been formally described, and to honor our collaboration with our Indonesian partners, all of these species have been named or derived using the Bahasa language, and also to honor the region in which these new Fusarium species were found. So then we decided to investigate the pathogenic potential of the most abundant Fusarium species found, which was Fusarium rheacanum. We did this by inoculating 2 isolates of Fusarium rheacanum into 16 8-month-old Acacia crassicarpa trees. After 30 days, the bark was peeled back at the inoculation site, and the lesion development was assessed. After 30 days, there was no external signs of disease on any of the inoculated plant hosts, and all that could be seen was, when you peeled the bark back, was these red lesions in the inoculated Acacia crassicarpa trees that were absent in the control. This, along with statistical analysis, suggests that this Fusarium species has a low level of aggressiveness. The role that this Fusarium species plays in the observed decline in the Acacia crassicarpa trees, if any, still needs to be determined, but it could be that this Fusarium species simply acts as a food source to its beetle associate. So here's just a closer look at the inoculations we did. So we believe that the observed decline in the Acacia crassicarpa could be due to a combination of the mass attack of these ambrosia beetles, as well as the repeated inoculation of these weakly aggressive Fusarium species. Now, although no observed ambrosia beetle infestations have been seen on eucalyptus plantations in Indonesia, these eucalyptus plantations are grown in close proximity to Acacia crassicarpa compartments showing heavy infestation with ambrosia beetle species. So we decided to do a preliminary pathogenicity trial using, again, the most abundant Fusarium species, Fusarium rudicana. So based on the results of this, the good news is that this Fusarium species appears to be even less aggressive on eucalyptus. So again, to reiterate, we do believe that the observed tree decline on these Acacia crassicarpa could be due to the combination of mass attack of beetles and the repeated inoculation of these weakly aggressive Fusarium species. However, these beetles could be contributing to the observed decline in these Acacia crassicarpa by aiding in the dispersal of other secondary, more aggressive plant pathogens. In the context of this disease paradigm, that could be Ceratocystis manganecans, which has been mentioned in previous talks. So they can do this. Professor Mike Wingfield touched on this morning. So they can aid in the dispersal by either creating entry wounds for the secondary plant pathogens to enter the host. These wounds could be visited by other insect vectors. They could vector other secondary plant pathogens, such as petidulid beetles, which have been associated with Ceratocystis manganecans, or simply that once a host is infested with Ceratocystis manganecans and they decide to bore into this host, they could then kick frass out, which you can see in the bottom two images. They could be inoculated with, for example, Ceratocystis manganecans, and this could therefore increase the inoculum load in the environment and aid in the dispersal of this pathogen. So in conclusion, in this study, we identified 2 Uralacea ambrosia beetle species as the most abundant ambrosia beetles infesting Acacia crassicarpa plantations in Indonesia. We identified 7 new Fusarium species that were associated with these 2 ambrosia beetles, and they have been formally described. Based on preliminary inoculation trials using the most abundant Fusarium species, we believe that this Fusarium species is not very aggressive, and that the damage caused to the Acacia crassicarpa plantations in Indonesia is most likely caused by mass attack of the beetles and repeated inoculation of these weakly aggressive Fusarium species. However, it should be noted that these beetles could play a role in aiding in the dispersal of other, more aggressive fungal pathogens, such as Ceratocystis manganecans. And with that, I'd like to thank my funding bodies, my collaborators, and thank you so much for listening.
Thank you, Ms. Kira. So we are ended our second speakers discussing on the ambrosia beetle species infesting the acacia. We go for the third presenter. Is it Ms. Havenga ready? Okay, yeah, okay, okay. Her research interests in the reproduction, population, genetics, and disease mechanism of fruits and plantation tree pathogens. She's active in the field of plant pathology, bioinformatics, and genomics, and uses basics and advanced molecular techniques to study and understand disease development. The development, biology, and pathology of plant pathogens. She obtained a PhD in genetics at University of Pretoria at the Forestry and Agriculture Biotechnology Institute, FABI. Okay, to date, she has 6 first author and 4 co-author publications. So I think you are ready, Miss Minute. Yes. Okay, the floor is yours, and probably you can share your slide, or the secretary will share the slides.
I will share my screen.
All right, please.
Okay.
Okay.
Can you see it fine?
Yeah, yeah, it's fine. And then please proceed. The
Okay, thank you so much for, well, good afternoon, firstly, everybody. And thank you so much for a lovely introduction. And thank you for giving me the opportunity today to present some of our research. So for this presentation, I'm not going to look at acacia, but rather another important plantation crop, eucalyptus. And I will present on the population biology of this global important eucalyptus foliar pathogen called Teratosphaera destructans. And then later in the presentation, I'm going to discuss a new tool that we developed for the rapid identification. The Teratosphaera destructans causes severe shoot and leaf blight, which leads to the defoliation and dieback of newly planted eucalyptus trees, especially those cultivated outside of their native environment. The severe symptoms includes defoliation, shoot and tip dieback, stunted trees, and it can even ultimately lead to the death of the trees. Under the moist conditions, asexual fruiting structures form below the stomata, and they exude these asexual conidial spores that then gets dispersed to neighboring trees. The sexual state of this fungus has never been found. So Teratosphaera destructans was first discovered and described from North Sumatra, Indonesia in 1995, and it subsequently spread rapidly to other tropical and subtropical eucalyptus growing regions, and it was most likely with contaminated seeds. It was reported in Thailand, in East Timor and Vietnam, in China, Laos, and most recently, it was reported to cause disease in Malaysia. In 2015, Teratosphaera destructans was discovered causing serious damage to eucalyptus plantations in South Africa. And this is the first case of the pathogen occurring outside of Southeast Asia. This pathogen has since spread to all Eucalyptus grandis regions in South Africa. But in 2007, Teratosphaera destructans was found in Australia. However, a 6-year-long survey was conducted in previously infected Australian sites, and the study concluded that the pathogen was misidentified and that Teratosphaera destructans have not been confirmed in Australia. Therefore, the origin of this pathogen is currently unknown. Teratosphaera destructans is currently limited to Southeast Asia and to South Africa. And even though this pathogen was discovered in 1995, very little is known regarding its diversity, its origin, or the dispersal pathways between the countries. Therefore, we investigated the genetic diversity, the recombination potential, as well as the global relatedness of 7 populations from both Southeast Asia and from South Africa. This was achieved by sequencing 3 Teratosphaera destructans genomes, and we used these genomes to develop and then to apply polymorphic microsatellite markers to a global collection of isolates. This collection consisted of isolates from 7 populations, of which 6 were from Southeast Asia. Specifically, we had populations from China, Thailand, a very small population from Vietnam, a population from Malaysia, and 2 populations from Indonesia. And then, of course, we also had a population from South Africa. These populations are geographically separated from one another, and we considered them to be independent. So this is what we found. We identified 38 genotypes, of which 37 were limited to their specific population. And if individuals are regularly moved between populations, we would expect more genotypes to be shared among our populations, which wasn't the case. We only, in fact, had 1 shared genotype between some of the populations, specifically several Thai isolates, all of the isolates from Vietnam and South Africa, as well as 2 isolates from North Sumatra. And these 2 isolates from North Sumatra were also coincidentally the 2 oldest isolates in our collection, because they were sampled back in 1995 from the original disease report, making this not only the only shared genotype, but it's also the oldest genotype in the collection. The Malaysian population had 20 unique genotypes, which was far more than any of the other populations. This was very surprising because new populations would not be genetically diverse, and it can even be clonal, as we saw for South Africa. And this is because young populations have undergone a bottleneck and have not had time to evolve. But this is clearly not the case in Malaysia. And then on the other hand, we can see that our 2 older populations in Indonesia had the 2nd most genotypes in their populations. And this would be expected for older populations. On the slide, we show the genetic distance among the genotypes in a minimum spanning network. So each node in the network represents a genotype, and the size of the pie is equal to the sample size. So the genotypes generally separated according to their sample regions. And since these areas are geographically separated from one another, our results indicated they were independent evolution after the pathogen was introduced into the different areas. The Malaysian genotypes did not intersperse with those from any of the other locations, and they were separated from all of the other populations by a larger genetic distance than typically found within the populations. So next we assessed the population structure, and we used two methods. And both of these methods assigned the individuals into the same 4 clusters. Again, we can see that our isolates grouped according to their geographical location, with the 2 Indonesian populations in cluster 1, populations with a shared genotype in cluster 2, the Malaysian isolates in cluster 3, and Chinese isolates in cluster 4. The Malaysian cluster had the highest levels of admixture, particularly with the Chinese cluster as well as the South African cluster, whereas all of our other populations had extremely low levels of admixture. The Malaysian population also had the highest genotypic diversity of 20%, which was nearly double the reported diversity of our older populations in Indonesia. And all of the other populations had low levels of genetic diversity. And this wasn't so surprising since 89% of the isolates belonging to these populations were clones. So then we wanted to investigate whether this fungus in each population predominantly reproduces via sexual or asexual reproduction. So why do we want to know this? Because populations that undergo sexual reproduction can produce individuals that have an increased fitness. It can attack a wider range of hosts, adapt better to changing environment, and all of this makes the fungus less responsive to disease management strategies. It is therefore very important to understand how this pathogen reproduces in each of the populations in the world. So in previous research, we identified that the pathogen has an outcrossing mating strategy, which means that 2 individuals of opposite mating types has to find one another in the field to be able to reproduce sexually. And we use this information and develop primers that target the conserved regions of these mating type genes. Now we can use these primers as well as the previous developed microsatellite markers to investigate the mating type distribution and the potential for sexual reproduction in each of our populations, and to conclude what is the dominant mode of reproduction. So if sexual reproduction regularly occurs in a population, a ratio of 1-to-1 mating types can be expected, whereas asexual populations are typically characterized by an uneven ratio of mating types. This is because, sorry, I just want to go back. So this is because the strong links between the alleles in asexual reproduction results in clonal populations with identical individuals, whereas sexual reproduction results in unique individuals. So now looking at our populations. The populations from Vietnam, Thailand, Indonesia, China, and South Africa all had uneven ratios of mating types, which suggests that sexual reproduction is unlikely in these populations. The South African population, that consisted of a single genotype, also has 1 mating type, further confirming that the South African collection of isolates are completely clonal. Sexual reproduction of this pathogen in South Africa is currently impossible without the introduction of the opposite mating type. If we look at Malaysia, this was the only population with an even ratio of mating types. This, together with evidence of random recombination, as well as the presence of multiple unique individuals and private alleles, indicates that sexual reproduction could be occurring in Malaysia. To summarize the reproduction results, all our investigated populations, except for Malaysia, had low genotypic diversity, uneven distributions of mating types, high levels of clones, and no evidence of recombination. This also suggests that the asexual state is the dominant mode of reproduction and dispersal of this pathogen in these areas. The Malaysian population also consisted of a high number of clones, which indicates that the asexual reproduction is still present in that population. And this is consistent with the fact that only the asexual state is known and is regularly found in heavily diseased trees. So for Malaysia, this population had the highest observed genetic diversity of any Stratospheric destructans population. It had an even distribution of mating types, evidence of recombination, as well as several private alleles. Our results suggest the presence of an active sexual cycle. These results were quite surprising since they contradict what is known for the older Stratospheric destructans populations, as well as for those closely related pathogens, all of which appears to reproduce only asexually. So sexual-driven evolution of this pathogen in Malaysia is quite concerning because this may lead to a more adaptable pathogen that is less responsive to disease management practices. So it brings us to the question, what is the origin of Stratospheric destructans? So native populations of fungal pathogens typically have a high genetic diversity and a greater number of private alleles compared to the introduced counterparts. And this is because introduced populations usually emerge only from a small number of individuals, where native populations have had a long period of time to evolve. So does this mean that the origin of Stratospheric destructans is in Malaysia? No, this is highly unlikely because this pathogen is very specific to eucalyptus, and there are no eucalyptus species native to the region. This indicates that all of our investigated populations represent non-native populations, and the origin of this pathogen remains unknown. It is, however, expected to be either Australia or East Timor. This is because the majority of eucalyptus species are native to Australia, and other teratospheric-causing leaf diseases of eucalyptus were first discovered in non-native plantations before they were finally observed in Australia. And this can also be the case for Stratospheric destructans. And then for East Timor, Stratospheric destructans has been reported on native eucalyptus species in East Timor, but we did not have any cultures to test this hypothesis. We found strong geographical structure, which suggests independent introductions into all of our sampled areas from an unknown source. However, two of our earliest collected isolates from North Sumatra share genotypes with isolates from Thailand, Vietnam, and South Africa. And it seems probable that North Sumatra could have been the source of disease outbreak for at least one of these countries. But importantly, the long distance spread of this pathogen between countries is clearly a consequence of human-mediated movement of infected plant material. And this highlights the need for much stricter quarantine measures when moving plant material to establish new plantations. If we look at the South African population, this was completely clonal, even though the collection consisted of isolates from multiple sampling sites and years. This indicates that there was a single introduction of the pathogen into South Africa. So I would like to end this presentation by looking at a diagnostic tool that we developed to rapidly identify 6 economically important Stratospheric pathogens. So as we saw in the presentation thus far, Stratospheric destructans are being introduced into new areas. And this is in the same for other important Stratospheric species. So this is the distribution of Stratospheric destructans, and if we add the currently known distribution of the other 5 economically important Stratospheric species, we can see that these species are not geographically distinct, meaning that there's more than one species occurring in some of the countries. So as you can see, it's getting more and more difficult to keep track of which species occurs in which areas. And to make matters worse, the identification of Stratospheric pathogens are quite time-consuming and expensive. And this is because the morphological characteristics and the initial disease symptoms of both the aggressive and the less aggressive species are very similar. Then this genus grows extremely slowly in culture. So as you can see, there's less than 2 centimeters of fungal growth obtained after 5 weeks. And this significantly delays the DNA extraction and identification. Then once we have quality DNA, identification to species level is not possible based on ITS sequence data alone, and we require 3 fungal barcoding regions. So due to time and cost involved, large-scale studies will typically only sequence a subset of isolates, and this leads to inaccuracies. So a rapid and cost-effective diagnostic tool is needed. So we assessed the developed microsatellite markers and developed a new stratosphere epicoida marker to distinguish among 6 stratosphere species. We wanted to use microsatellites because they have had a higher amplification success for small amounts of DNA. And we were now also able to look at 13 loci using only 2 PCR reactions. So this is a snapshot of the results. Highlighted in the table are the specific loci that did not amplify for each of the species. And then based on the size of the amplicons, the microsatellites can accurately distinguish among our 6 stratosphere species, and it can be used as a diagnostic tool, especially in large-scale disease surveys, inoculation studies, or to monitor for new stratosphere destructants introductions into populations that already exist or into new areas. So I would like to thank our funding bodies for their invaluable contribution for us to be able to do this work. And then if you would like to know more about the work presented here today was published in 4 publications shown on the screen. And then I'll also be available for questions after the talk, or you can reach me on Twitter. Thank you so much.
Thank you so much. Okay, this is good to know that Malaysia has sexual reproductions and highest diversity, even ratio of mating types and so on, as compared to other countries which is asexual reproductions. Okay, we come for the Q&A section. Probably we can see from the Q&A chat room. Do we have any questions for the first questions? Okay, I think this is for the, sorry. Okay, can, the speakers check the Q&A, Q&A room to see for the, to read for the question. Okay,
I think there's a question from
Nugara. Yeah, the details is from Ms. Kira.
Okay. Pa Mohammed, I'm not entirely sure what's in East Kalimantan. So I don't know if it's the same Ulacea ambrosia beetles, but if it's from the pruned Acacia crassicarpa, and if it's on basically a dead piece of the wood, it could potentially be a variety of ambrosia beetle species, because ambrosia beetles naturally are attracted to dead or decaying wood. So I wouldn't be able to give you a clear answer, but if you could get a sample of the beetle and maybe get a picture of it, it would be easier to identify. You still need maybe phylogenetic DNA sequence to be 100% sure, but they're quite easily distinguished based on morphology. So you could maybe send a picture and I could potentially identify it. Sorry, I don't know if that helps.
Okay, thank you, Ms. Kira. And then, yeah, there is so far there is question in the chat. Is it the same question, Mr. Muhammad Nugra? Identify as well. Yeah, I think the same questions. Okay. Probably I have one question with, to Ms. Havinga. In your opinion, what are the reasons that Malaysia has high genetic diversity in the non-population of this pathogen? Yeah.
There can be multiple reasons, and I think we need to go back and research again. It can either be that the samples for the specific population all came from a single plantation. So that's also quite concerning that the high genetic diversity all originated from a single plantation. But it can be that the plant material that was used to establish the plantation had previous infected Stratosphaera destructans with different genotypes already. And then there might be the environmental conditions in the plantation. It's just conducive for sexual reproduction increasing, because we did see sexual reproduction is occurring. So it's definitely multiple introductions and then potentially sexual reproduction, just increasing the diversity. So it would be interesting to go back to nurseries and to see what is the propagation material looking like.
Okay, thank you. I tried to find anybody probably raise their hand. Okay. Probably I have one question on the biosecurity. Dr. Simon, are you sure that this biosecurity system appropriately address the current and future biosecurity challenge? Yes.
Yeah, it's a very good question. We are trying to take small steps, I guess, is the scope of the project. So as I said, we designed the network to have something to do. So it's not a network that just exists without context. So the high-risk site surveillance, as I kind of pointed out, draws in lots of other aspects of and highlights what things you need to do to have a good biosecurity system. So we expect there are going to be lots of difficulties. Diagnostics is a big problem for everybody. So how do we get good diagnostics on the things that we find? What's the capacity of each country to be able to share data? I mean, this is really important that we be able to share data. But that's also quite a political, can be quite a political issue as well. So they're the kind of things. But there certainly seems to be a need to start somewhere, and if we can get better cooperation across the region than there is currently, then we would have done at least some part of the job. So also, as I said, we actually have quite a big policy and social science component to the project. So I'm an entomologist, not a social scientist, but we do have a social scientist employed on the project, and so that will be his job is to try and draw the network together and to work on those, emphasising those policy issues, such that each country really does value forest biosecurity as something that they should be investing in the longer term. So not sure if that answers your question, but it's small steps to start with. Yeah.
Probably there is a continuous question according to that things. Probably which is the most highest risk significant to the current and future biosecurity for Malaysia, and then probably if that the most highest risk, and is there any mitigation approach to handle that?
So one of the very first things we'll do in each country is start putting together pest risk lists for forest pests. But certainly with the increased emphasis on eucalyptus, there are a lot of eucalyptus pests moving around the world. So, I mean, one was emphasised with Nam's paper today with this new pathogen emerging in Sumatra. That would certainly be something that everybody needs to start looking for immediately in the region. But there are other eucalypt insect pests that have been moving around the world. So Malaysia already has, I think, Leptocybe invasive, the gall wasp. There was another gall wasp in the region in Indonesia, Ophthalmimus, 2 species. And then there are some others that have been moving around and established elsewhere in the world, like South Africa and Brazil with the bronze bug, Themastocorus. It's been a huge pest, not currently in Asia or Southeast Asia. So yeah, but certainly for Malaysia to start looking at but something like the disease that Nam talked about is maybe one of the most important ones to look for immediately. Yeah.
It's a great suggestion, Simon. I believe that there is authorities will do take note on your suggestion, and they will do further improve the current situation to mitigate the problem later. I think Mr. Kira keep busy answering, texting the answer. Then so far no person is raised their hand to give a questions, and then we can end our session now. Any comment again from the speakers? Or want to add on? And no questions from the floor, but everyone can email to the presenters as been given. Yeah, you can email because the address can be get it. Your email address can be get from the abstracts that been shared by the Ms. Shetlin. I think that's all for this session. Ms. MC, hand over to you.
Okay, ladies and gentlemen, we will be having a short break for 15 minutes. We will resume the session at 3:20 p.m. 3:20 p.m. Thank you.
Thank you. Thank you,
Thanks,
all the
everybody.
speakers. Thank you, everybody.
Thank you.
Thank you for all the attention.
Welcome back, ladies and gentlemen. To proceed with our final part of session 3, I would like to invite Professor Dr. Kalima to lead the final session. Dr. Kalima, please.
Thank you, Mr. MC. Very nice introduction. Okay, we go for the last session under risk management scope of this conference of third IUFRO Acacia Conference 2021. I would like to introduce you Mr. Manuel Asby. Is around? Is here, right? Okay. Okay, Mr. Manuel is a logistic manager at Forest Solution Malaysia, Sdn Bhd. He took his Bachelor of Science in Forestry awarded in Göttingen, Germany. Currently a master student with the University Putra Malaysia, studying and splitting in Eucalyptus pelita plantation log. Again, 23 years of work experience, whereof 20 years in Malaysia. And then work experience is harvesting, log yard operation, plantation establishment, nursery plant production, and as well as R&D. So I believe that Mr. Manuel is ready. Okay, I can see you there. Okay, all right, okay. Want to sharing your screen? Oh, all right, please. Manuel. Hello, is it Secretariat ready with the slide? And then Mr. Manuel, are you? Okay, great. Okay, Mr. Manuel. No. Can you hear us?
Yeah, I can hear you now.
That's good. Okay, please proceed. And the floor is yours. Is it fine? Please. Mr. Manuel, can you present your slide? We can hear you. Mr. Manuel? Sorry, Secretariat, any, all right, I'm ready. So we couldn't get him. So Manuel, are you ready? Or you can pass to Professor Irene. Audio system. Okay, is it Professor? So is it you need time to
Prof. Kalina?
Yeah.
Can we proceed to the next speaker?
Okay, next presenter. Okay, Professor Irene, are you here?
Yes, I'm with everybody.
Okay, here you are. Professor Binus is an associate professor and research leader in the Tree Protection Cooperative Program, TPCP, and the Forestry and Agricultural Biotechnology Institute, Fabi, University of Pretoria. Her research focuses on the taxonomy, phytogenetics, and population biological of fungal pathogens that infect forestry. Okay, and then Professor holds leadership position in South African Genetics Society, SAGS, International Union of Forest Research Organization, IUFRO, and on the Executive Council on the International Mycological Association, IUNA. How about your slide? Are you going to share a slide?
Yes, I'll share the slides.
Okay, that's great. Again, show please. All right.
Okay, great.
Great,
Is that
so
working well?
you're ready.
Right, good afternoon, everybody. It's a real pleasure for me to be able to join this conference from abroad and in South Africa. And I would really like to thank the conference organizations for making this possible. The work that I'll be presenting today is really a long-time collaboration that we've had in FABI in South Africa and with Saba Softwoods in Bahad, especially with Yanni Yaparudin, as well as Rezwan Wealth, who actually came to FABI to do his master's degree with us. Our general aims in this work is really to characterize ceratocystis species, but also to determine the risks that they might pose to certain tree species, in this case, especially acacia species. Just see where I can advance. So we have been dealing with ceratocystis in South Africa for a really long time because we too have an acacia species that is affected by the disease, and we use it in plantation forestry. And this is Acacia mearnsii, also known as the black wattle. It has similar symptoms that everybody here is familiar with, in the fact that it produces rapid wilting and dieback of trees. And then in the middle here, you will see gummosis that is produced by the tree and depressions and cankers. And this is really the tree trying to protect itself from the pathogen. And if you cut open into the wood and you expose the internal symptoms, you will see that there's typical streaking that can be observed in these tree species. The pathogen that causes this particular disease is known as ceratocystis albopundus, and it's unique in that it actually has a white base, where other species have a dark base. And you can see this up at the top here. And from the genetic characterization that we've done in population genetic studies, we found that this pathogen is actually native to the African continent and South Africa, whereas its host, Acacia mearnsii, is exotic and it comes from Australia. So this is something to look out for in the future where other people are planting acacia species. But acacia species in other parts of the world are also being affected by ceratocystis, and especially in Southeast Asia, in Indonesia and Vietnam, for example. It is, however, caused by a different ceratocystis pathogen, but the symptoms are the same. So in this picture, you will see Acacia mangium in Indonesia that is already at the end stages of the infection, where there is total tree death. This plague of infection has caused extensive and large-scale death, especially again in Indonesia. In fact, so much to the extent that it has caused the total collapse of the Acacia mangium industry. But as you would have heard from the previous talks, Mahathir has given us a glimmer of hope that this could potentially be re-established. But this problem really started in 2003 and has subsequently changed the face of forestry, where eucalyptus species, particularly Lepelita, has been grown now as a replacement species to replace this Acacia mangium. The pathogen that causes this disease, however, is known as Ceratocystis manganecans. But it's important to note that Ceratocystis manganecans is not host-specific to acacia species. In fact, we first described the species causing infection on mango trees in Oman and subsequently in Pakistan. Here you can see the tree death that is occurring, the gummosis from the wounds on the trees and the cankers, and then as well as xylem discoloration. But the problem here is that it wasn't only on the mango trees, it also moved to the native trees in Oman and Pakistan. For example, here we've got Dalbergia sishu, which is the shisham tree, and it also infects Prosopis cinerea, which is garth. And these are native tree species, so it's really concerning. And with genetic characterization that we did, we actually found it's the exact same clone. It's a single clone that infected mango that caused 100,000 death of mango trees is now also infecting these native tree species. So it's of concern, but it doesn't stop there. This pathogen also infects pomegranate. You can see extensive dieback here on the pomegranate tree, as well as really extensive discoloration. We found it on eucalyptus in many places in the world. You can see some canker depressions there, very characteristic star-shaped staining of the wood as well. But what's really concerning is that during the last 3 years, for example, there have been numerous other hosts that have been reported to be infected by Ceratocystis manganicans. And you will see they are the loquat, jackfruit, duku, bulletwood. And the concern here is that these tree species are planted across different areas. They're also in different locations. They've been reported in Indonesia and Vietnam and Pakistan, for example. So as Simon was talking about looking at biosecurity risks, the fact that this manganicans is infecting such a large, wide host range is concerning and something that we really need to take note of. The extent of this host rate is not known because these new reports are quite recent, guava as well. So the pathogen in all of these cases is either reported as being due to Ceratocystis manganicans or to Ceratocystis fimbriata. And I will discuss a little bit why there's confusion regarding what we're actually calling these pathogens in some future slides. But back to Malaysia. Of course, we've got Ceratocystis canker in Malaysia, and it's also causing the same symptoms and the same kind of disease and extent that we've been finding in Indonesia. Here it basically started in 2011, but again, driving the change to be where a eucalyptus is planted instead. So we have Ceratocystis on Acacia mangium in Malaysia, but we also have another Ceratocystis species that is in Malaysia, and this is one caused by Ceratocystis fimbriata, so that strange name comes back up again. And this infects sweet potato. The symptoms on sweet potato is what we call a black rot. And you can see this discoloration here in the tuber, and there is some fungal growth that is occurring. So these are two basic pathogens in Malaysia, Ceratocystis with these kind of names. The importance with sweet potato in Southeast Asia and Malaysia, of course, is that it is a very, it's a crop of major importance to the communities over there, but it's an excellent source of carbohydrates and it's good to grow economically. There are different kinds of varieties, and a lot of these varieties do get infected with Ceratocystis fimbriata. So we've got these basic two Ceratocystis species. So what is Ceratocystis? For those of you who don't know, it is a fungus. It's in the order Microascales, and this is a picture of its sexual stage where it has the base here, generally that's black, and this top piece over here is the spore that carries all the ascospores, so a really good source of inoculum. It belongs to the family Ceratocystidaceae, and this particular genus is Ceratocystis. Now, if we're looking at the phylogenetic clades of Ceratocystis and what is actually in there, these species can be generally divided into 4 major clades, and this depends on where they've really been found and locations in terms of the type species. And we're interested in what we call the Latin American clade. In this clade, there are species that cause aggressive tree death. For example, we've got the Ceratocystis manganicans in there, but we've also got Ceratocystis platani causing death of plane trees, as well as the new species that is being described in Hawaii causing rapid ohia death there. But now, depending on your species concept, you might recognize that there are 42 different species of Ceratocystis in the genus, or you might recognize that there are 37 species. And so what do I mean by this? And I'm just going to spend a moment to explain this because it's important when we come to naming things. So we have different species concepts. And say now this is our fungal pathogen and it is on the road to divergence, which means it's starting to speciate. If you are looking at a phylogenetic species concept, it's more or less right at the beginning where speciation is occurring. And so your genetic differentiation between these populations is starting. And in here, you can recognize a phylogenetic species by these clades being monophyletic. So it's based on genetic fixed polymorphisms between species. And as time goes on, you might then find that these 2 species that are starting to diverge are starting to also experience other things like ecological differences, such as they might be in new niches or new hosts. And as time goes on, then these two species might develop morphological differences. And these are your morphological species. So they might differ in the way that they look, for example. And during all of these processes of phylogenetic, ecological, and morphological species boundaries, there is this barrier to gene flow that is really starting to develop. In some species, it is already quite developed early on, and in others, potentially not so much. So in this case, there could be hybridization taking place between the different species. If you believe in the biological species concept, then at this stage, there is complete reproductive isolation, which means that the two individuals of different species, they cannot mate and produce progeny. So this is where we get our 42 species versus our 37. So what does this really mean for us in terms of Ceratocystis species? So if we're looking at our phylogenetic clade, and we're concentrating on this Latin American group in blue that I showed you previously, if you are looking at a phylogenetic species concept, then all of these separate blocks we identify as individual species. So you will have Ceratocystis eucalypticola, manginecans that we're interested in. You will also have a different species as termed Ceratocystis fimbriata, different species based on a combined phylogenetic approach. This fimbriata in the phylogenetic species concept only occurs on sweet potato. So now if you're looking at the biological species concept, at this node here, all of those other individual species that were recognized in the phylogenetic species sort of collapses, and they all become known as one species. And in this case, all of them here would then be known as Ceratocystis fimbriata. And why would we even think about having this difference? Well, that's because Ceratocystis fimbriata and manginecans, if you put them on a plate, you can actually cross them. They produce in this hybrid zone here, fertile progeny. And this is why, in some cases, people believe that they're actually all the same species. However, we've only managed to cross these species in a laboratory situation. And although we get viable offspring, they are not fit. In other words, they die, and we actually struggle to maintain them in terms of keeping them alive in the lab. So whether this hybridization or recombination is happening in the field, we don't know. It seems unlikely because we don't have any evidence of it in thousands of isolates that we've basically looked at. So if it is occurring, it might not be fit enough. So these are the basic biological species concepts that come into play when we're describing Ceratocystis and where the confusion lies in the literature. So based on the fact that we've got these two different species of Ceratocystis that we're concerned about now in Malaysia, and the fact that we've got these different species concept, we have raised certain questions. So we would like to know, firstly, are the pathogens causing Ceratocystis wilt on Acacia mangium and black rot of sweet potato in Malaysia the same fungus? So basically, is it the same thing? To do this, we collected some samples, and then we used a multi-gene phylogenetic approach to characterize these species. Then we have other questions. Is the pathogen causing the wilt on Acacia mangium equally aggressive on Acacia auriculiformis and crassicarpa? And this is kind of questions that have actually been answered in many of the previous talks. So there's good foundation for this. And thirdly, because sweet potato is commonly cultivated in close proximity to acacia plantations, we wanted to know, is there a risk of the sweet potato pathogen then jumping host to infect any of these acacia species? Bearing in mind that I've just shown you that Ceratocystis manginecans can infect a wide host range of species. So do the sweet potato pathogens pose a risk? And in order to answer this question, we did artificial inoculations. Our sample collection was on Acacia mangium in 3 different provinces in Malaysia, in different regions. And these are the kind of symptoms that we were looking for in terms of the Acacia mangium. And these black spots over here with this white fungal growth starting is where we did isolations, and either directly or through carrot baiting technique. And then Ralph did big excursions, particularly in Sabah, where he then went and collected infected sweet potato tubers, specifically looking for these black depressions that you can see here, which is this black rot, and that little white over there indicates that the fungus is already starting to grow there. So from this material, we did carrot baiting if we didn't take it directly from the wood material. And here you can see some ascospores producing. We got clean cultures, we did DNA extractions, and we did a phylogenetic comparison based on 4 gene regions. Notice the ITS was not included here. And this is something that Sri brought up earlier, and perhaps we can talk about this in the discussion. In terms of artificial inoculations, the isolates that we got from Acacia, we then inoculated 2 different isolates onto the 3 different host species, Acacia mangium, crassicarpa, and auriculaformis. And we did the same then with the sweet potato fungus. We just chose 2 isolates and also did the inoculations. And then of course we had a control as well. So our collection sites in Malaysia was just where we managed to find at that stage the acacia infected as well as sweet potato, and these are the different sites as indicated with the orange dot. You can see that site Tawau having both of the hosts that we're looking at. When we were looking for symptoms on Acacia mangium, the symptoms varied. So you could look for cankers such as on the left-hand side over here, or depressions, or characteristic symptoms also included the foaminess and gummosis. And when you cut into either one of these, if you saw the streaking appearance over here, that's exactly what we were looking for, that xylem discoloration, and isolations were made from those. Now, taking a phylogenetic approach and sequencing a whole bunch of these isolates, only a few are represented in these trees, we basically got this distinction between the 2 different hosts, whereas all the isolates in the blue that were isolated from Acacia mangium grouped together in a clade, and this one we know is Ceratocystis manginecans. It also groups with other isolates from Acacia mangium in Indonesia, as well as the mango in Oman. All of the isolates, and this is over 100, that we isolated from sweet potato grouped within the Ceratocystis fimbriata clade, and these were mainly from regions within Sabah. So basically we have 2 very distinct species in 2 very distinct clades on their respective hosts. So what does this really mean in terms of the risks to acacia? So if we take the Acacia mangia trees and we inoculate them with the respective pathogens, on the left here, we can see an inoculation with Ceratocystis manginecans, and extensive lesions have formed 4 weeks after inoculation. Whereas if you look at the inoculation with the sweet potato fungus, there was nothing. And if we just zoom in, here you can still see a little bit of the plug, but definitely no infection has taken place. Whereas if you're inoculating with manginecans, you're getting this extensive lesion forming, sometimes so large that it's up to 2 meters high in some of the other inoculations that were done. So if we take a closer look at just what's happening then across the 3 different acacia species, the bars in blue indicate the results when inoculated with Acacia mangium on Acacia mangium. And you can see these lesions are quite significant, but with the sweet potato fungus, there was nothing, so no lesion forming. We had similar results when inoculated onto auriculaformis, where it was definitely pathogenic to a lesser extent though than Acacia mangium, but again, nothing happening when we inoculated Ceratocystis fimbriata, and exactly the same results when inoculated onto Acacia crassicarpa. So definitely showing that the sweet potato pathogen is not infecting acacia. So in conclusion, there are 2 very distinct species considered in the study, and it's clear that what is affecting acacia and causing wilt on acacia is due to Ceratocystis manginecans. And the isolates that we are finding on potato causing black rot is only Ceratocystis fimbriata. We know that all 3 species of acacia are susceptible to infection by Ceratocystis mankinicans, but of course, Acacia mangium is significantly the most susceptible. And based on our results, we also know now that Ceratocystis fimbriata poses no threat to the acacia plantations and that a host jump is unlikely. So in this case, if you're planting them next to each other, there should be no cross-infections. So this is okay. I would like to acknowledge members of the Borneo Forestry Cooperative, especially Sabah Softwoods, where the experiments were conducted, David Bowden, who coordinated the work, as well as Mahadir for assistance with the inoculations. And thank you everybody for listening.
Thank you, Prof, for your insightful presentation of this pathogen. And then we do hope that we can hear from Mr. Manuel, but unfortunately, he has a problem on the audio system. The CAT Secretariat tried to fix to upload the pre-recorded video later in the next session or tomorrow session. We go straight to the Q&A session for this presentation by Prof. Barnes. So any question from the floor? I can see any raised hand from Mr. Ani. And then I can start for the ball rolling. Prof, is it confirmed that this pathogen, this fimbriata, was completely no threat to the acacia? It's really, as you said, it's very joyful. But then it can be said that previous presenter also highlighted about this issue as a threat to the acacia. So there is a finding towards the end of today, but then to balance the information that we learned earlier in this morning.
So this is an exceptionally important question and an exceptionally important issue that we are facing when we're looking for identification of Ceratocystis species. Generally, the ITS region has been used to characterize the species, and in the past this has actually worked really, really well, and it is considered a barcoding region to identify most pathogens. But unfortunately, when it comes to Ceratocystis mankinicans, the ITS region contains multiple copies. And depending on which one you're sequencing, you might get a different result. So, and one individual isolate, when you sequence it, could actually have 2 different sequences, one putting that into mankinicans and one putting that into something else. And this is where the confusion comes in with using that gene region to do characterization. So if you're only using that, which is what some people are doing for identification purposes, you kind of find yourself in a difficult position because you may have it in 2 clades, but it actually represents one individual species. And so when it comes to Ceratocystis, at the moment, we recommend that anybody who's describing things for the first time and on a new host, that you need to adopt a multi-gene phylogenetic approach to make sure that you're correctly identifying the isolate. So ITS is not really a good gene region to use for any of these classification systems. And that is why if you don't use the ITS, you can distinguish quite clearly between the mankinicans pathogen and that one causing fimbriata. So if you do believe, if you are adopting the biological species concept and everything is being called fimbriata, what people are doing now is that they're assigning haplotypes to the different lineages, which means that you can at least still identify where things are coming from and you can make connections. The problem is if you don't do that and you just say everything's fimbriata, then people get really confused as to what pathogen is infecting what and on what host.
Hardianto, do you have any interesting question to Prof? Oh. I can see Mr. Shafiari raise your hand. So are you have a question for Prof? No. Anyone else from the floor? Questions? If not, we can end the sessions for the risk management scope. I believe that everyone has gained so much insightful information Thank you to all participants' attentions on this risk management scope. And then apologies for any inconvenience caused by our sides. For the Mr. Manuel, we noted that you're going to present your output or your presentation tomorrow by pre-recorded. We do hope everyone has a fruitful session with nice and full of information of the research finding for the whole day today. Please stay tuned for the fourth session of this conference. Will be moderated by another person. I think that's all I can over to the this MC, Mr. MC. Okay.
Thank you so much, Professor Dr. Kalina Abdan, for your contributions as a moderator in this session. Thank you as well to all speakers for this afternoon session, Dr. Simon Lawson, Ms. Kira Lin, Dr. Minet Havenga, and also Professor Dr. Irene Barnes. Without further delay, distinguished participants, ladies and gentlemen, we have come to our fourth session on high value added timber product. I am most pleased to welcome the moderator for this session, Ms. Annie Ting. Ms. Annie is the Chief Executive Officer of Sarawak Timber Association. She holds a bachelor's degree in business administration, majoring in finance from Middle Tennessee State University, USA. Since starting her career in the Sarawak Timber Association, STA, in September 1989, she has worked her way to become the Chief Executive Officer in STA. She brings with her a wealth of experience from over 30 years of working closely with STA members, relevant government agencies, and international organizations to advance and promote forestry and tropical timber trade, as well as deal with policy issues. Please let us welcome Ms. Annie Ting. Ms. Annie, please.
Thank you, MC. Afternoon, everyone. First of all, I'd like to congratulate Forestry Department Sarawak for organizing this timely and important webinar. And also thank the organizing committee for inviting me as a moderator for this session. This session, we have 6 presentations. This afternoon, we will welcome 3 invited speakers. And first, I hope Professor Dr. Razak Wahab, you are around. Are you around? Because you'll be the first one to present for this afternoon session. Is on the high value added timber product. We actually move away from the management of the plantation, and now we are toward the downstream. So Professor Dr. Razak, are you around? Before I introduce the speaker, because we are ahead of the time, just want to make sure that the speaker is around. Secretariat, would you be able to identify whether the speaker is around? Anyone from Secretariat?
Hello, Miss Annie.
Yes.
Can we start from Dr. Sik Hui Sing, the third speaker?
The third speaker. Okay, can. The third presentation, it will be on the, let me see. On the commercial production of quality cane-dry acacia dimension salt. Dr. Sik Hui Sing, currently a research officer and a head of wood drying laboratory at the Forest Research Institute, Malaysia. She joined FRAME after attending her Bachelor of Science in Forestry, majoring in wood industry from UPM. She got her doctorate in material science from National University of Malaysia. Her research area include environment friendly processing of timber, particularly in high pressure treatment of plantation timbers, and developing high temperature timber treatment technology based on the local innovation. And she has received a few awards, including the IUFRO Outstanding Doctorate Research Award at the 24th IUFRO World Congress held in Salt Lake City, Utah in USA in 2014. I will pass the floor to Dr. Seth. You have 20 minutes for your presentation. Dr. Seth, the floor is yours. Is Dr. Seth? Oh, there you are. Okay.
Hi.
Hi.
Just came back from a small break. Yeah.
Okay.
Thank you very much, Miss Anity. So yeah, let me share my presentation.
You may start your presentation whenever you are ready.
Sure, sure. Right. Okay. Okay, I'm ready. Let me check. Okay. Good afternoon. Yes, good afternoon to the participants. A very good afternoon to all the participants of the third IUFRO Acacia Conference 2021. First of all, I would like to thank the opportunity to share with you some of our projects that we carry out in Sarawak. And the title of my presentation today is the Commercial Production of Quality Kiln-Dried Acacia Dimension Stocks. Basically, this project is a collaboration with the a local Sarawak-based company, and the main projects of this project is to develop a drying treatment system for particularly for acacia, and also this system is also suitable to dry other low to medium density timber. The main project, as I mentioned earlier, is to fully utilize the plantation resources in Sarawak and mainly the acacia. And probably this figures has been doubled since a few years back. And the project was started in about 4 years back. And the aim of this project is basically to develop an efficient drying system to treat acacia. Mainly to overcome some of the major conventional drying problem in acacia. And let's look at some of the drying problem in acacia. Basically, you have very long drying time, And basically, some of the producer took about probably 6 to 8 weeks to dry acacia from green to a kiln-dried timber that are suitable to develop into products. And then this results in very low turnaround time. And also we have found that there's a lot of, when the timber is dried, we discovered there's a lot of uneven moisture content in the timber, which render the timber not suitable to produce into quality products. And sometimes this will lead into severe drying defects such as collapse of the timber and the internal tracks, as you can see in the diagram, where this kind of internal tracks will severely degrade the integral structure of the timber. And let's give you an example, an overview. Yes. Let's give you an overview of how a commercial drying plant looks like. So basically, a drying plant consists of the boiler system, and this boiler will supply sufficient steam supply to the kiln dryer. So what is a kiln dryer? It's basically a drying chamber consists of the hardware such as the heating system, and we have some ventilation system as well to dry and condition the timber. And throughout the important thing here is that the system must have good control over the process. And the process here means that the operator that dry the timber. So you have the good equipment, but what about the people that manage the timber? So the personnel has to have the know-how to dry properly the timbers. And of course, different timbers have different drying behavior. So we must learn the characteristics of the timber before we apply a certain recipe into the timber. So in all is to effectively remove the water from the timber so that we don't degrade the timber. Next, let's give you an overview what's the activities that we've been carried out on the ground. So I can see here, this is typically a log yard before the acacia logs being transported for further processing. And you go to what we call the primary processing in the sawmilling section. And after that, the timber will well stacked into the box stack timber and we load into the kiln. And after drying, the timber will be loaded. And of course, there will be a lot of QC will be going on. Like the most important is to check the moisture content. And also and the other secondary processing is that the timber was further dressed into the suitable thickness that as required by the clients before the timbers are being well packed into the bundles to be ready for shipment. Next, we also carry out some log recovery studies. Basically, we know that the planted acacia in Sarawak, is mainly for supply the pulp and paper production. So I think what we understand from the local producer is that throughout the plantation period, there's lack of silviculture treatment. So the timber is actually comparatively smaller than those forest timber. And we did some ground studies and found out that the log diameter for the acacia ranging from 6" to 13" for 10 to 13 years old acacia. But nonetheless, we think that there's still a lot of the value of the timber to be salvaged. That's why our aim is to produce, using advanced technology to potentially use the small log diameters and put it into a high value timber products. And this is the system, the projects that on the ground, where we actually with the the opportunity that's given by our industry collaborator, we actually work from design of the kiln itself until of course the designs that were put into the kiln is actually what we have in mind is that we want to best treat the acacia timber, because acacia timber compared to other timbers is a bit challenging to treat because of the wet moisture pockets and other issues such as warpings being a juvenile timber. And also with that, we actually managed to establish an efficient kiln drying system, which consists of 4 high-performance dryers. And each of this kiln drying system is actually about 100 cubic meter of timber capacity. So you can imagine the one that we have in markets now probably up to about 50 tons or about 70 cubic meter. So this is quite big, it's quite a big capacity compared to the conventional kiln that is in the market. So basically, with the cooperation rendered by our industry collaborators, we managed to do a very upscale R&D straight from our lab to the commercial, the industrial scale. So we are thankful for the opportunity for us to develop such system with our industry collaborator. And also both parties have actually managed to file this invention we have filed an IP for this invention, and the details are, of course, we are willing to disclose when the innovation is ready to be shared with the rest of the. And throughout the implementation of this project, 2 utmost goals in both the research teams and our industry collaborator is that first is to how best to dry the timber in the shortest time possible, but without compromising the quality of the timber. In other words, is that we want to dry the timber fast, but without actually degrade the integrity of the timber, and also we want to get the best quality out of the timber. And all this is very important, that we strike a balance between getting the fast kiln throughput and getting the best quality. And of course, the higher you have lower production costs. And let's look at the comparison between the current conventional method that has been applied generally, and as compared to the accelerated drying methods that developed with our project teams. So basically, you can see here is that the current conventional methods require 2 processes, where first they will air dry first the timber. So the air drying probably took about 3 to 4 weeks, depends on the thickness of timber. And then the timber will be subject to second stage of drying, which is into the kiln. And normally they apply the temperature not more than 8 degrees Celsius, whereby using our method of the accelerated drying method, we only apply the single process, whereby we dry the timber from green condition up to the desired moisture content. So we apply the temperature up to about 150 degrees Celsius. Of course, when you look at the time, the higher temperature actually helps to speed up the process. So from the normal processing of the acacia drying from 6 to 8 weeks, now we only took about 14 days, about 2 weeks to complete the drying of the timber from green to kiln dry. And of course, with shorter drying time period, you have lower stock holding, meaning that you can better manage the the procurement of the raw material. So you don't have to stock up a lot of your lots in the lot yards. So that basically save you a lot of the stock holding costs. And also, besides that, we found that the timber quality of our kiln-dried acacia is slightly enhanced, most probably due to the higher temperature that we applied, which actually helps to enhance the timber in terms of the mechanical properties, and also some to some extent the physical properties of the timber. And basically, how do we actually to gauge that we have actually properly dried the timber? As you can see here, the timber, the specimens here is actually taken at different drying periods. And with effective drying methods, you can actually eliminate or minimize the incidence of wet pockets. So it's not how difficult it is to dry a timber, it's whether you know how to dry a timber. So no matter how recalcitrant is the timber, as long as you know the behavior of the timber, you can actually apply the proper drying techniques to overcome the problems of the inherent problems of the drying issues. And Another qualitative analysis that we use or a standard way to gauge whether your timber has been properly dried is of course to use the standard oven dry methods to check the moisture content of the timber. So basically the approach that we use is that we put sample boards in the kiln and then we check the moisture content before and after drying. So basically we do the sectioning of the timber to actually look further into whether the timber is dry evenly throughout the surface regions as well as the core regions, to make sure that there's no wet pockets right in the middle of timber that actually will give you a lot of problems when the timber is subsequently used for processing. And when you look at the moisture content of the timber before drying, these are the series of the sample boards that we put into the timber. So when you see that's each of the sample is, we have a pair of the moisture content specimens. So as you can see there, some of the timbers within the timber might have very close moisture content, like the S7. You can see this is actually from the same timber. Whereas when you compare another timber, S12, you can see there's a large variation within the timber itself, meaning that one end is actually 70% of moisture, the other end might be more than 100%. So that's why acacia is a very challenging timber to dry mostly because of the moisture content of the timber. And also when we further look into the sectioning of the moisture content, we can see that, for example, the S1B2, you can see that in the core, you have much higher moisture content. And this also explains why that most of producers are having some problem, because when they air dry the timber, after a week or two, they will see a lot of issues of collapse the timber even before going into the kiln, because the large moisture gradient will happens during the air drying period. So this will actually give you a lot of issues when going to the kiln because of the large moisture gradient. And when we look into after drying, what about after drying? So using our method, we actually cut the same timber board after drying, and we found that the average final moisture content of the whole batch is around perhaps 7 to 9%, and this is actually is evenly, you can see within the boards is actually quite evenly between 1 or 2%, which is acceptable in most scenarios. And when we look further into each of the pieces, okay, we also found that the difference of moisture content between the core and the surface is actually just within 1 or 2%, which is actually within the general permissible range of the moisture content. And besides doing the moisture content test, we also do other physical properties tests, such as the density and the shrinkage. And also we also carry out mechanical properties tests. And also we check on the working properties of timber, whether it's harder to work against the conventional or otherwise. But from the feedback of our industry, we found that generally the timber is quite good to work with. And also we also test on other things like bonding tests when they do lamination. Especially, these are the tests that we carry out to ensure that the timber is actually very suitable to produce into other products that actually fetch higher value, that actually brings more returns with the investments that they make. And so these are some of the packagings that are already ready to be shipped out. And of course, these are each of the timber is actually going through very stringent moisture content checks using moisture meter, and each of the pieces are being checked to ensure that there's no very discrepancy in the moisture content, because the buyers also are very strict on the specification. So the general rule is that the moisture content of the batches doesn't exceed 14%. So we managed to actually bring down the timber, the moisture content a bit, because the timber, when you leave, when you expose, it will reabsorb after kiln drying. So basically, is to make sure that the reabsorption of the timber, the moisture doesn't actually affect the timber qualities in a later stage. So with that, we thank you for your attention to share with us today. And also we would like to take the opportunity to thank our industry collaborator, Isbat Lumbers Sdn Bhd, which is located in Bintul, Sarawak, for giving us the opportunity to work on the ground and to actually carry out R&Ds that can actually help the industry to further value add their timbers. With that, I thank you for your attention.
Yeah, thank you, Dr. Sheikh. Actually, your presentation is very clear and straightforward. And I think that with this, we'll address a lot of the industry concern. Because in order to go value added, I think that KD is a very important component. So that the products that, especially using the solid timber, they have confidence in the go down to the further value added products like the furniture. So I think that we will leave the question and answer during the session, question and answer session. So thank you, Dr. Sheikh, for your presentation.
Thank you, Madam Chair.
With this, I would like to maybe go back to the first speaker, first invited speaker. Are you ready? Professor Dr. Razak. If not, how about the second speaker, Dr. Ilama Ran? Are you around? Again, I need Secretariat to help. If the two invited speaker is not around, anyone?
Prof. Razak. Prof. Razak is there.
Professor Dr. Razak. Okay, good.
Can you hear me or not?
I can hear you very
Can you hear me?
I can. We can hear you. Okay. Maybe I will introduce the speaker before I pass the floor to you, Professor. Okay. Professor Dr. Razak is currently working with the University College of Technology, Sarawak. And he is the head of the Center of Excellence in Wood Engineer Products. He have many years of experience with FRIM, and his research area cover wood science and technology, wood composite, wood chemistry, engineer wood products, and wood utilization, and have published 165 papers and also 39 international proceeding paper and 12 book. Very experienced professor, and currently involving in the engineer bamboo research under the Sarawak State Government Bamboo Industry Development Project. With this, Professor, I will pass the mic for your presentation. You can proceed with your presentation whenever you are ready.
All right, can you see my slide here?
Yeah, can.
All right. Okay, thank you, Miss Ann Ting, the CEO of Sarawak Timber Association. My fellow researcher, ladies and gentlemen. Okay, the title of my paper is on this acacia engineered wood product. Okay, Sarawak is rich with bio-natural resources, especially timber and other non-timber forests produced. The focus on the timber industry now is on upstream product like sawn timber, plywood, veneer, particle board, and so on. To generate more income to the state, there is a need to shift to the downstream industry, especially the furniture industry and the development of the timber housing component industry. The Sarawak Corridor of Renewable Energy, or SCORE, in 2014 have declared that timber-based industry as one of the priority industry to bring the basic commodity timber production into manufacturing high value component and finished good. Engineered wood, the modification process in manufacturing of wood product which possess better and improved properties than in its raw form. In the engineered wood, the wood and adhesive are mixed with the strand, particle, fiber, or veneer or board of the wood to form a composite product. Engineered wood were designed according to the national or international standard specification. Okay, Sarawak in 2018 has managed to produce wood and timber product value at 5.4 billion Malaysian ringgit in export earning. The production mainly focus on upstream industry such as sawn timber, charcoal, briquette, veneer, plywood, laminated board, wood molding, wood pallet, wood chip, particle board, MDF, and laminated flooring. Furniture that can generate high income have not been really established yet in Sarawak and still need to be imported from Peninsular Malaysia or overseas. The wood and adhesive will mix with the strand, fiber or veneer or board to form this composite board. Okay, with the permanent forest and forest plantation covering 6 million hectares and 2.8 million hectares respectively, Sarawak have high potential to increase the downstream product. Okay, this paper discuss the application of cultivated acacia wood as potential material for laminated veneer lumber, composite board, cross-laminated board, and wood trimmer. Okay, this is the Center of Excellence for Wood Engineered Product, or the acronym is CWEP. Okay, CWEP was established in 2017 with the aim in enhancing the development of downstream wood product industry in Sarawak. The objective of this CWEP is to develop advanced engineered wood product from local wood species, especially the planted timber in Sarawak. Number two, to produce engineered wood product of lesser known species with low matured age for housing construction and as well for furniture. Number three, to fully utilize the wood yield with producing a wood composite product.
Professor, Doctor.
Yes.
Interrupt. Can you put your presentation in the slide more? Currently, you are. I think the participant will not be able to see the slides clearly.
Oh,
maybe you can click. You have to book. No, it's not in the slide presentation. More. Maybe you have to press. Is it you control or the secretariat?
Can the organizing committee share the slide with me or not?
Who is organizer? Is the organizing committee?
I have a copy of my slide.
Oh, okay.
We can share. Prof, can you please stop sharing your slide?
Okay. This is the first time I'm using this, you know. Okay, I stop sharing. All right. Yeah.
Thank you, Secretariat.
Okay, can we go to slide 7? Okay, after this, yeah, this is the short-term KPI. Number one, in-house development to conduct in-house training in the development of the engineered wood product. Number two, to acquire new skill among members of the CWEP. Number three, to act as the leading research center for the wood-based product in Sarawak. Number 4, conducting training for this local timber-based industry. And then for the long-term KPI, assisting in increasing the export of engineered wood product in the timber industry in Sarawak. Number 2, assisting in the utilization of engineered wood product as one of the main building material and for construction. Number 3, assisting in increasing the income of the local community and also generating money for the state government. Okay, can we go to next slide, please? All right, these are the strategic planning. Okay, we are going to focus on analysis or assessment in producing and utilizing the new engineered wood product or the new product design, and then conducting laboratory work or field work on the subject of interest. This include introduction of new engineered product or product design through exhibition, and then getting feedback from the industry for us to evaluate our planning. Evaluating of the new wood species or new wood product for its marketability and marketing, and then training the member in the Center of Excellence and from the industry regarding to the new product design. Can we go to next slide, please? Okay, this is the business plan of the Center of Excellence in wood engineered product. The main activities for the acacia wood development, they are divided into several stages. This include the harvesting of the acacia wood from the existing plantation around Sarawak. The CWEB will act as the center in providing the R&D and training services on the development of the acacia raw material into wood engineered product. And this include the production of trimble, cross-laminated board, wood laminated, and composite. The engineered wood product are going to be assembled into semi-finished product, such as this engineered wood flooring, house component, structure, and furniture. Okay, to realize this center, we will work closely with the other main agency in Sarawak, such as the Sarawak Timber Industry Development Corporation, Sabah Forestry Center, and also with the STA. Okay, can we move on to the next slide, please? Okay, some of the engineered wood products are well established in Malaysia, and the existing type of the engineered wood product are such as plywood, fiber board, particle board, and laminated timber. The plywood, okay, these are manufactured using the peel veneer and bonded together with selected adhesive. The fiber board and particle board are produced using the particle board size of wood and bonded with adhesive. And the laminated timber, okay, they are processed based on layer of wood pieces and glued together to form a larger pieces of product. Can we go to the next slide? Okay, the engineered wood product are also widely produced using softwood and hardwood species. However, research about the manufacturing using the acacia species are still new, and information need to be discussed further. Okay, among the type of wood engineered product that can be introduced are the laminated veneer, the cross-laminated timber, trimble, parallel strand, and laminated strand wood. The engineered wood product, they have the possess properties of high strength, good stability, and they are multi-function material they are good, especially in construction purposes. Okay, the combination of acacia and high performance adhesive will give a great impact on the manufacturing of this wood engineered product. Can we go to the next slide? Okay, focusing on wood shrimble manufacturing, it can be produced using the wood pieces and also from this veneer. Okay, this slide shows the variation of the wood shrimble that are produced. Okay, the wood shrimble, okay, they can be produced using a veneer, and this veneer can be arranged in such a way that it will give some sort of this ring growth. On the appearance side, shrimble products are suitable for luxurious furniture with an artificial wood grain array and growth ring. On the property side, the shrimble product have good strength and they are more durable for heavy structural construction. Can we go to next slide? Okay, these are some of the engineered wood manufacturing process as presented in the figure. The engineered wood manufactured based on three primary material that are used, mainly the plain sawn, the veneer, and the sawdust. Each material form represent the different identity of product that are going to be developed. Okay, the ready material form, they are normally they are dry and then they are preserved before spreading adhesive for the formation purposes. The residue were then molded or stacked according to the arrangement that has been specified. Next, the forming were cured under cold or hot press. Okay, finally, the finished product were conditioned before they are ready for packaging and labeling. Okay, can we go to the next slide? Okay, a few types of wood composite and engineered product were introduced according to their sharing purposes. This include this glue lamp, cross-laminated timber, laminated vinyl lumber, OSB, wood plastic composite, MDF, and plywood. Can we go to next slide? Okay, this slide show the glue laminated manufacturing using small and short pieces of wood. Okay, they are bonded together to form longer pieces of product. Normally, we will use a finger joint in order to bond the material together. Okay, this slide show the laminated timber that are used for the structural engineered wood. Okay, they are constituted of layer of dimensional stable lumber that are bonded together, forming a more durable and having a better strength. Okay, can we proceed to the next slide? Next slide, please. Okay, the laminated veneer lumber is an engineered wood product that use multiple layer of thin wood that are assembled with adhesive. Okay, this slide show the cross-laminated timber, not the veneer that I mentioned just now. It is the wood panel, okay, that are formed by gluing pieces of solid timber together. They are mainly used for the structural material Okay, can we go to the next slide? Okay, this is a oriented strain board. The investment for this is a bit too high. I would not recommend it to be introduced here. But anyway, this OSB are used extensively, especially in Europe. Okay, this oriented strain board is a type of engineered wood, okay, similar to the particle board. It is formed by adding adhesive and then compressed layer of wood strand into a specific orientation. The OSB is a material which favorable mechanical properties that make it particularly suitable for load-bearing application, especially in construction. And the most common uses are in shading, in wall, flooring, roof decking, and furniture. Besides, the OSB is stronger. They are much stronger compared to the plywood in shear. Can we have the next slide, please? Okay, this show the sandwich structure composite. This sandwich structural composite is a special class of composite material that is fabricated by attaching two thin but stiff skin to this lightweight but thick core. The core material is normally low strength material, but it higher thickness provide the sandwich composite with the high bending stiffness with its overall low density. Okay, despite the thickness of the core, the sandwich composite, they are light and have a relatively high structural strength. Okay, this composite have a spatial structure which affect good thermal insulator properties. The sandwich panel are used in aerodynamic road vehicle, ship, and civil engineering. Can we go to the next slide? Okay, this slide show the wood plastic composite. Okay, this wood composite, wood plastic composite. They are composite material made from wood fiber or wood floor, and thermoplastic such as polythene, polypropylene, polyvinyl chloride, or polyacetic acid. Okay, the wood plastic composite sandwich board consists of wood polymer composite skin and usually have low density polymer core, which lead to a very effective increase in the panel rigidity. Okay, this WPC, they are normally used for this outdoor deck floor, but it is also used for making a railing, fan, landscaping, timber cladding, and siding park benches, molding and trim, as well as for making window and door frame, and also for the indoor furniture. Okay, can we go to the next slide? Okay, this I think everybody know the medium density fiberboard. Okay, medium density fiberboard. Okay, is an engineered wood product made by breaking down the hardwood or softwood residual into wood fiber. Okay, then they are combining with wax and resin binder. Okay, forming into panel by applying high pressure and temperature. This MDF, okay, they are generally possess stronger and denser properties compared to the particle board. Okay, can we go to the next slide? Okay, this is plywood. Okay, everybody, I think everybody know plywood. Plywood is a material, okay, manufactured by thin layer or plies of wood veneer that are glued together, which adjacent layer having their wood grain, okay, rotated up to 90 degree or to the other. A typical plywood panel has face veneer of higher grade, okay, than the core veneer. Okay, so far the use of Acacia hybrid, it will, okay, easily produce the best quality veneer, which can be used for decorative purposes. Plywood for the general use, and particle board, okay, made from chip, can also use the conventional plywood. Okay, can we go to next slide? Okay, okay. Treatment of acacia wood. At the moment, we use preservative to treat our wood, but here I would consider the application of wood heat treatment as it can act as this preservative in enhancing the durability of the wood. Normally, we will suggest the heat treatment, okay, to apply temperature at above 160 degrees Celsius, which can significantly improve the dimensional stability of the wood. It increases the wood biological durability and increase the dimensional stability. Okay, this wood heat treatment has been widely used in the wood industry, especially in Europe, which is resulted from the systematically fundamental study, and due to its good environmental effect. Okay, can we go to the next slide? Okay, engineered wood product, okay, are used in a variety of application, from home construction to this commercial building to the industrial product. The product can be used for joists and beam that can replace steel, okay, in many building projects. Okay, mass timber, okay, is a group of building material that can replace concrete, okay, assembly. Can we go to the next slide? Okay, this building, okay, they are actually superior compared to the application of steel. It is a renewable and sustainable resources material, okay, but they have and it has unlimited design opportunity, and often create a carbon neutral environment. Can we go to the next slide? Okay, the picture on the right, okay, not this one. Yeah, the next slide. This is similar to the previous slide. Okay, these are some of the major application of this engineered wood in Europe. Okay, we can see they use laminated timber that are compressed together, forming a larger portion of this timber frame. Okay, can we go to the next slide? Okay, these are some of the application of the engineered wood in small or medium-sized house. We would recommend the application of the engineered wood for the construction of this small and medium-sized house in Sarawak and also in Sabah. All right, can we go to the next slide? Okay, these are some of these laminated timber from Acacia. I took this picture from this timber exhibition that was held in Kuching, I think in 2019. You can see, okay, these are some of the products. Okay, they're forming very heavy design. Alright, can we go to the next slide? Okay, these are some of this laminated floor or component for parquet manufacturing. You can see the parquet are manufactured by at least 2 or 3 components to make it the floor more dimensional stable. Okay, can we go to the next slide? Okay, this is the Acacia house that was built in frame in 1998. They are manufactured from solid and also laminated Acacia mangium. Can we go to the next slide? Okay, these are some of the defects that occur. I think probably because of this improper application of the preservative. After such a time, you can see the leftover attack of the termite on the wood pieces. I think this one, this problem can be overcome if we use heat treatment. Okay, can we go to the next one? Okay, this is a bridge that was built in frame using heavy hardwood, such as balau and red balau. Okay, both of the beam were constructed using different this adhesive. I think it was sponsored by this Exo Nobel Adhesive and Jawad Manufacturing. Okay, the performance of the wood and adhesive and as well as the finishing, I think they are being monitored regularly. The first bridge, I think, was constructed or was installed in 2006. And this is I think the third bridge that was built. All right, can we go to the next slide? Okay, this is the cost-limited timber house that was built recently in frame. This is the type of house that we plan to build with some modification. I think probably the size of this house is just like a container, 10 by 10, sorry, 10 by 10 and then by 20, which is probably not having good space, you know. Most probably we are going to do double the space. Can we, no. Slide 34. Okay, this one. Okay, all right, then it's okay. This is actually, it is an office that was constructed using this glue laminated at this MTIB office in Johor. I think this office was constructed in 2012, and it was officially launched by the former Prime Minister. Okay. Okay, I think that's all, ladies and gentlemen. Thank you.
Thank you, Professor, for your very detailed introduction to different engineering wood products and also the usage. Okay, I think before I open up to the question and answer, and once again, I would like to check whether Dr. Emil Maran around, because he is now based in India. So, are you in the group, Doctor? I guess maybe not in the group. Secretary, would you want to confirm that he is not in the group? Then we can proceed to Q&A.
Dr. Elamaran, can you please raise your hand? I think he's not in. I think we'll proceed to Q&A.
Okay, can. Thank you. Now we will spend 15 minutes for the Q&A. I see that Dr. Shet have started to answer the chat group question. Anyone would like to have any maybe question to the professor and also Dr. Shet? Any question? Or their presentation is very clear with a lot of information. Maybe I will ask Dr. Shet while others thinking of their question. For your this drying protocol, is it only for acacia or you can open up to other planted species? So maybe eucalyptus or other species that as I said, in our plantation.
Thank you for the question, Madam Chair. Basically, the system is actually you can actually dry timbers from low to medium timbers, because normally heavy hardwood we don't dry because it's very recalcitrant to dry. So any timber that's ranging from low to medium density will be suitable for kiln drying. And in fact, we actually did some eucalyptus drying in West Malaysia, but because West Malaysia we have very little planted eucalyptus, so we have yet to have the opportunity to dry eucalyptus in a larger scale. So hopefully we can work with some of our counterparts in Sabah, Sarawak to actually do more research on this eucalyptus drying. Yes.
I think in your chat you did mention that you are now currently pending for the IP. When do you roughly know that you will get the IP?
Basically, the IP will need time to get published. So once the documents has been published and for review, then we are able to open up more on the technical sides of the research.
Okay. Yeah. Currently, would you provide any maybe if there is any query in detail of the drawing about the protocol, would you be able to maybe welcome the contact?
Yeah, sure. Because we've been working very closely with industry. In fact, for the last few years, we also work with our counterparts in Sabah on acacia, but that is on the conventional steam heated acacia. So in Sarawak, we applied more on drying and heat treatment. So basically, acacia we've been doing quite a lot of localities, even up from Indonesia. Also, we tried some acacia from Indonesia and also from Peninsular Malaysia. So we found that actually acacia timber from all from different provinces has different characteristics. So some of the quality in, for instance, in Sabah, we found that it's quite good quality. So it also depends on the provinces. I mean, the locality that the timber is planted. So we welcome anyone that wants, would like to collaborate with us on, further research into acacia drying impacts other species like eucalyptus and paulownia, which is the upcoming timber. That would be great to work with. Yes.
Okay. Thank you.
Can I ask a question or not?
Sure. You can.
Sorry. Actually, I'm writing a book on this acacia. Okay. Have you worked on acacia hybrid or not? Do acacia hybrid pose problem in drying?
Yes, and in fact, we have opportunity to work with some like auriculaformis and some hybrids and also some superbulb. So all this different acacia actually has different drying behavior. So but the thing is that we have very little opportunity to work with hybrid or the superbulb. So most probably we can have more materials to work with. Yes.
Okay. Anyone from the participant? I see from the question and answer, Dr. Wahab, okay, wonderful work. I wonder if you and your team have worked with young thick timber, and if so, what is your opinion on their marketability in this part of the world? Professor, that is for you.
Can you rephrase the question again?
Okay, I will. Okay. He actually would like to know whether you and your team have worked with young thick timber. If so, what is your opinion on their marketability in this part of the world? Especially whether your team work on the young thick timber, thick T-E-A-K, thick.
Okay, you mean young age timber?
Yeah, thick timber. Yeah, that's right.
Okay. Normally for this engineered wood, we would recommend the application of young timber. The timber that we harvest from the forest. Okay, they are matured. Probably they are more than 30 or maybe 40 years. The one that we recommended are probably between 10 or 12 years old. They have low density, but if you produce veneer and glue them together, they will give better properties compared to the timber that we harvest from this forest. Okay, so far I work on the properties and utilization, but not on the marketing yet. We kind of hope in 1 or 2 years time, we can produce a model house that are going to build using this planted timber species in Sarawak in order to promote the application of this engineered wood to the local people.
Yeah. Okay. Because so far I always hear for professor member saying that the engineering wood product, the investment is very high, but the products don't really attract a good price as compared to what it has to be invested. What is your comment?
Yeah, I agree with you. The investment for this engineered wood, okay, product, okay, they are very high. But I kind of recommend we utilize this LVL, the laminated veneer lumber, and then the cross-laminated timber. Okay, we can use this existing machine in order to bond the pieces of veneer or timber together to make it this wood plank. Yeah, I think I've read some article. Okay, there's one company in Indonesia, I think it's in Jakarta. They produce wood plank by compressing this veneer together. You know.
Okay. Okay. So far, you do any research on this? Maybe the LVL or the cross-laminated timber.
Yeah, we are in the early stage of producing this. Actually, like I told you, right now we are doing 5 projects. Mostly these projects are not big, you know.
Okay.
Hopefully, we can embark on a bigger project early next year. No? We are going to get. We are applying for a greater or bigger grant research. You know.
Okay. Noted. Okay, I see another question. Let me see. This one is from Hamza. How is the cost of between the conventional treatment and the accelerated treatment with the improve of acacia treatment? Can it be increased the price of wood? I guess this is for Dr. Shet.
Okay, thank you for the question. Actually, to compare the, it's totally different equipment. The etc. drying requires high performance drying system, meaning that whatever you use is actually has higher resistance